Chapter 29 Vertebrates 29.1 Chordates. By the end of this section, you will be able to do the following. Describe the distinguishing characteristics of chordates. Identify the derived characters of craniates that sets them apart from other chordates. Describe the developmental fate of the notochord in vertebrates. The vertebrates exhibit two major innovations in their evolution from the invertebrate chordates. These innovations may be associated with the whole genome duplications that resulted in a quadruplication of the basic chordate genome, including the Hox gene loci that regulate the placement of structures along the three axes of the body. One of the first major steps was the emergence of the quadrupeds in the form of the amphibians. A second step was the evolution of the amniotic egg, which, similar to the evolution of pollen. Figure 29.1 Examples of critically endangered vertebrate species include a. The Siberian tiger, Panthera tigris, b. The mountain gorilla, Gorilla barangay, and c. The harpy eagle, Harpia harpyja. The harpy eagle is considered near threatened globally, but critically endangered in much of its former range in Mexico and Central America. Credit A. Modification of work by Dave Pape. Credit B. Modification of work by Dave Proffer. Credit C. Modification of work by Howie Ard. Introduction. Vertebrates are among the most recognizable organisms of the animal kingdom, and more than 62,000 vertebrate species have been identified. The vertebrate species now living represent only a small portion of the vertebrates that have existed in the past. The best-known extinct vertebrates are the dinosaurs, a unique group of reptiles, some of which reach sizes not seen before or after in terrestrial animals. In fact, they were the dominant terrestrial animals for 150 million years, until most of them died out in a mass extinction near the end of the Cretaceous period, except for the feathered theropod ancestors of modern birds, whose direct descendants now number nearly 10,000 species. Although it is not known with certainty what caused this mass extinction, not only of dinosaurs, but of many other groups of organisms, a great deal is known about the anatomy of the dinosaurs and early birds, given the preservation of numerous skeletal elements, nests, eggs, and embryos in the fossil record. Chapter Outline 29.1 Chordates 29.2 Fishes 29.3 Amphibians 29.4 Reptiles 29.5 Birds 29.6 Mammals 29.7 The evolution of primates and seeds in plants, freed terrestrial animals from their dependence on water for fertilization and embryonic development. Within the amniotes, modifications of keratinous epidermal structures have given rise to scales, claws, hair, and feathers. The scales of reptiles sealed their skins against water loss, while hair and feathers provided insulation to support the evolution of endothermy, as well as served other functions such as camouflage and mate attraction in the vertebrate lineages that led to birds and mammals. Currently, a number of vertebrate species face extinction primarily due to habitat loss and pollution. According to the International Union for the Conservation of Nature, more than 6,000 vertebrate species are classified as threatened. Amphibians and mammals are the classes with the greatest percentage of threatened species, with 29% of all amphibians and 21% of all mammals classified as threatened. Attempts are being made around the world to prevent the extinction of threatened species. For example, the Biodiversity Action Plan is an international program, ratified by 188 countries, which is designed to protect species and habitats. Vertebrates are members of the kingdom Animalia and the phylum Chordata, figure 29.2. Recall that animals that possess bilateral symmetry can be divided into two groups, protozomes and deuterostomes, based on their patterns of embryonic development. The deuterostomes, whose name translates as second mouth, consist of two major phyla, Echinodermata and Chordata. Echinoderms are invertebrate marine animals that have pentaradial symmetry and a spiny body covering, a group that includes sea stars, sea urchins, and sea cucumbers. The most conspicuous and familiar members of Chordata are vertebrates, but this phylum also includes two groups of invertebrate chordates. Figure 29.2 Deuterostome phylogeny. All chordates are deuterostomes possessing a notochord at some stage of their life cycle. Characteristics of Chordata animals in the phylum Chordata share 5 key characteristics that appear at some stage during their development: a notochord, a dorsal hollow, tubular, nerve cord, pharyngeal gill arches or slits, a post-anal tail, and an endostyle, thyroid gland, figure 29.3. In some groups, some of these key characteristics are present only during embryonic development. The chordates are named for the notochord, which is a flexible, rod-shaped mesodermal structure that is found in the embryonic stage of all chordates and in the adult stage of some chordate species. It is 780. Chapter 29, Vertebrates access for free at openstax.org. Strengthened with glycoproteins similar to cartilage and covered with a collagenous sheath. The notochord is located between the digestive tube and the nerve cord, and provides rigid skeletal support as well as a flexible location for attachment of axial muscles. In some chordates, the notochord acts as the primary axial support of the body throughout the animal's lifetime. However, in vertebrates, craniates, the notochord is present only during embryonic development, at which time it induces the development of the neural tube and serves as a support for the developing embryonic body. The notochord, however, is not found in the postembryonic stages of vertebrates. At this point, it has been replaced by the vertebral column, that is, the spine. Visual connection. Figure 29.3 chordate features. In chordates, 4 common features appear at some point during development: a notochord, a dorsal hollow nerve cord, pharyngeal slits, and a post-anal tail. The endostyle is embedded in the floor of the pharynx. Which of the following statements about common features of chordates is true? 1. The dorsal hollow nerve cord is part of the chordate central nervous system. 2. In vertebrate fishes, the pharyngeal slits become the gills. 3. Humans are not chordates because humans do not have a tail. 4. Vertebrates do not have a notochord at any point in their development. Instead, they have a vertebral column. 5. The endostyle secretes steroid hormones. The dorsal hollow nerve cord is derived from ectoderm that rolls into a hollow tube during development. In chordates, it is located dorsally to the notochord. In contrast, the nervous system in protostome animal phyla is characterized by solid nerve cords that are located either ventrally and/or laterally to the gut. In vertebrates, the neural tube develops into the brain and spinal cord, which together comprise the central nervous system, CNS. The peripheral nervous system, PNS, refers to the peripheral nerves, including the cranial nerves, lying outside of the brain and spinal cord. Pharyngeal slits are openings in the pharynx, the region just posterior to the mouth, that extend to the outside environment. In organisms that live in aquatic environments, pharyngeal slits allow for the exit of water that enters the mouth during feeding. Some invertebrate chordates use the pharyngeal slits to filter food out of the water that enters the mouth. The endostyle is a strip of ciliated mucus-producing tissue in the floor of the pharynx. Food particles trapped in the mucus are moved along the endostyle toward the gut. The endostyle also produces substances similar to thyroid hormones and is homologous with the thyroid gland in vertebrates. In vertebrate fishes, the pharyngeal slits are modified into gill supports, and in jawed fishes, into jaw supports. In tetrapods, land vertebrates, the slits are highly modified into components of the ear, and tonsils and thymus glands. In other vertebrates, pharyngeal arches, derived from all 3 germ layers, give rise to the oral jaw from the first pharyngeal arch, with the second arch becoming the hyoid and jaw support. The post-anal tail is a posterior elongation of the body, extending beyond the anus. The tail contains skeletal elements and muscles, which provide a source of locomotion in aquatic species, such as fishes. In some terrestrial vertebrates, the tail also helps with balance, courting, and signaling when danger is near. In humans and other great apes, the post-anal tail is reduced to a vestigial coccyx, tailbone, that aids in balance during sitting. Link to learning. Click for a video discussing the evolution of chordates and 5 characteristics that they share. Click to view content. https://www.openstax.org/l/chordate_evol 29.1 Chordates 781 Chordates and the evolution of vertebrates. 2 clades of chordates are invertebrates, Cephalochordata and Urochordata. Members of these groups also possess the 5 distinctive features of chordates at some point during their development. Cephalochordata. Members of Cephalochordata possess a notochord, dorsal hollow tubular nerve cord, pharyngeal slits, endostyle, thyroid gland, and a post-anal tail in the adult stage, figure 29.4. The notochord extends into the head, which gives the subphylum its name. Although the neural tube also extends into the head region, there is no well-defined brain, and the nervous system is centered around a hollow nerve cord lying above the notochord. Extinct members of this subphylum include Pichia, which is the oldest known cephalochordate. Excellently preserved Pichia fossils were recovered from the Burgess shales of Canada and date to the middle of the Cambrian age, making them more than 500 million years old. Its anatomy of Pichia closely resembles that of the extant lancelet in the genus Branchiostoma. The lancelets are named for their blade-like shape. Lancelets are only a few centimeters long and are usually found buried in sand at the bottom of warm temperate and tropical seas. Cephalochordates are suspension feeders. A water current is created by cilia in the mouth, and is filtered through oral tentacles. Water from the mouth then enters the pharyngeal slits, which filter out food particles. The filtered water collects in a gill chamber called the atrium and exits through the atriopore. Trapped food particles are caught in a stream of mucus produced by the endostyle in a ventral ciliated fold, or groove, of the pharynx and carried to the gut. Most gas exchange occurs across the body surface. Sexes are separate and gametes are released into the water through the atriopore for external fertilization. Figure 29.4 Cephalochordate anatomy. In the lancelet and other cephalochordates, the notochord extends into the head region. Adult lancelets retain all 5 key characteristics of chordates: a notochord, a dorsal hollow nerve cord, pharyngeal slits, an endostyle, and a postanal tail. Urochordata. The 1,600 species of Urochordata are also known as tunicates, figure 29.5. The name tunicate derives from the cellulose-like carbohydrate material, called the tunic, which covers the outer body of tunicates. Although tunicates are classified as chordates, the adults do not have a notochord, a dorsal hollow nerve cord, or a postanal tail, although they do have pharyngeal slits and an endostyle. The tadpole, larval form, however, possesses all 5 structures. Most tunicates are hermaphrodites. Their larvae hatch from eggs inside the adult tunicate's body. After hatching, a tunicate larva, possessing all 5 chordate features, swims for a few days until it finds a suitable surface on which it can attach, usually in a dark or shaded location. It then attaches via the head to the surface and undergoes metamorphosis into the adult form, at which point the notochord, nerve cord, and tail disappear, leaving the pharyngeal gill slits and the endostyle as the 2 remaining features of its chordate morphology. 782 Chapter 29, Vertebrates access for free at openstax.org. Figure 29.5 Urochordate anatomy. A. This photograph shows a colony of the tunicate Batrylloides violaceus. B. The larval stage of the tunicate possesses all of the features characteristic of chordates: a notochord, a dorsal hollow nerve cord, pharyngeal slits, an endostyle, and a post-anal tail. C. In the adult stage, the notochord, nerve cord, and tail disappear, leaving just the pharyngeal slits and endostyle. Credit. Modification of work by Dan Blackwood, USGS. Adult tunicates may be either solitary or colonial forms, and some species may reproduce by budding. Most tunicates live a sessile existence on the ocean floor and are suspension feeders. However, chains of thaliation tunicates called salps, figure 29.6, can swim actively while feeding, propelling themselves as they move water through the pharyngeal slits. The primary foods of tunicates are plankton and detritus. Seawater enters the tunicate's body through its incurrent siphon. Suspended material is filtered out of this water by a mucus net produced by the endostyle and is passed into the intestine via the action of cilia. The anus empties into the excurrent siphon, which expels wastes and water. Tunicates are found in shallow ocean waters around the world. Figure 29.6 Salps. These colonial tunicates feed on phytoplankton. Salps are sequential hermaphrodites, with younger female colonies fertilized by older male colonies. Credit. Oregon Department of Fish and Wildlife via Wikimedia Commons. Subphylum Vertebrata. Craniata. A cranium is a bony, cartilaginous, or fibrous structure surrounding the brain, jaw, and facial bones. Figure 29.7. Most bilaterally symmetrical animals have a head. Of these, those that have a cranium comprise the clade Craniata, Vertebrata, which includes the primitively jawless myxini, hagfishes, Petromyzontida, lampreys, and all of the organisms called vertebrates. We should note that the myxini have a cranium but lack a backbone. 29.1. Chordates 783 Figure 29.7a Craniate skull. The subphylum Craniata, or Vertebrata, including this placoderm fish, Dunkleosteus sp. are characterized by the presence of a cranium, mandible, and other facial bones. Credit. Stevok 86, Wikimedia Commons. Members of the phylum Craniata, Vertebrata display the 5 characteristic features of the chordates. However, members of this group also share derived characteristics that distinguish them from invertebrate chordates. Vertebrates are named for the vertebral column, composed of vertebrae, a series of separate, irregularly shaped bones joined together to form a backbone, figure 29.8. Initially, the vertebrae form in segments around the embryonic notochord, but eventually replace it in adults. In most derived vertebrates, the notochord becomes the nucleus pulposus of the intervertebral discs that cushion and support adjacent vertebrae. Figure 29.8a Vertebrate skeleton. Vertebrata are characterized by the presence of a backbone, such as the one that runs through the middle of this fish. All vertebrates are in the Craniata clade and have a cranium. Credit. Ernest V. Moore. Taken at Smithsonian Museum of Natural History, Washington, D.C. The relationship of the vertebrates to the invertebrate chordates has been a matter of contention, but although these cladistic relationships are still being examined, it appears that the Craniata, Vertebrata are a monophyletic group that shares the 5 basic chordate characteristics with the other 2 subphyla, Urochordata and Cephalochordata. Traditional phylogenies place the cephalochordates as a sister clade to the chordates, a view that has been supported by most current molecular analyses. This hypothesis is further supported by the discovery of a fossil in China from the genus Hycoeuella. This organism seems to be an intermediate form between cephalochordates and vertebrates. The Hycoeuella fossils are about 530 million years old and appear similar to modern lancelets. These organisms had a brain and eyes, as do vertebrates, but lack the skull found in craniates. This evidence suggests that vertebrates arose during the Cambrian explosion. Vertebrates are the largest group of chordates, with more than 62,000 living species, which are grouped based on anatomical and physiological traits. More than one classification and naming scheme is used for these animals. Here we will consider the traditional groups Agnatha, Chondrichthyes, Osteichthyes, Amphibia, Reptilia, Aves, and Mammalia, which constitute classes in the subphylum Vertebrata, Craniata. Virtually all modern cladists classify birds within Reptilia, which correctly reflects their 1 Chen, J.Y., Huang, D.Y., and Li, C.W., an early Cambrian craniate like chordate, Nature 402, 1999, 518–522, doi. 10.1038, 990,080. 784 Chapter 29, Vertebrates access for free at OpenStax.org. Evolutionary heritage. Thus, we now have the non-avian reptiles and the avian reptiles in our reptilian classification. We consider them separately only for convenience. Further, we will consider hagfishes and lampreys together as jawless fishes, the Agnatha, although emerging classification schemes separate them into chordate jawless fishes, the hagfishes, and vertebrate jawless fishes, the lampreys. Animals that possess jaws are known as gnathostomes, which means jawed mouth. Gnathostomes include fishes and tetrapods. Tetrapod literally means four-footed, which refers to the phylogenetic history of various land vertebrates. Even though in some of the tetrapods, the limbs may have been modified for purposes other than walking. Tetrapods include amphibians, reptiles, birds, and mammals, and technically could also refer to the extinct fish-like groups that gave rise to the tetrapods. Tetrapods can be further divided into 2 groups, amphibians and amniotes. Amniotes are animals whose eggs contain 4 extraembryonic membranes, yolk sac, amnion, chorion, and allantois, that provide nutrition and a water-retaining environment for their embryos. Amniotes are adapted for terrestrial living, and include mammals, reptiles, and birds. 29.2 Fishes. By the end of this section, you will be able to do the following. Describe the difference between jawless and jawed fishes. Discuss the distinguishing features of sharks and rays compared to other modern fishes. Modern fishes include an estimated 31,000 species, by far the most of all clades within the vertebrata. Fishes were the earliest vertebrates, with jawless species being the earliest forms and jawed species evolving later. They are active feeders, rather than sessile, suspension feeders. The agnatha, jawless fishes, the hagfishes and lampreys, have a distinct cranium and complex sense organs including eyes, that distinguish them from the invertebrate chordates, the urochordates and cephalochordates. Jawless fishes. Superclass agnatha jawless fishes, agnatha, are craniates representing an ancient vertebrate lineage that arose over 550 million years ago. In the past, hagfishes and lampreys were sometimes recognized as separate clades within the agnatha, primarily because lampreys were regarded as true vertebrates, whereas hagfishes were not. However, recent molecular data, both from rRNA and mtDNA, as well as embryological data, provide strong support for the hypothesis that living agnathans, previously called cyclostomes, are monophyletic, and thus share recent common ancestry. The discussion below, for convenience, separates the modern cyclostomes into the class Myxinae and class Petromyzontida. The defining features of the living jawless fishes are the lack of jaws and lack of paired lateral appendages, fins. They also lack internal ossification and scales, although these are not defining features of the clade. Some of the earliest jawless fishes were the armored ostracoderms, which translates to shell skin, vertebrate fishes encased in bony armor, unlike present-day jawless fishes, which lack bone in their scales. Some ostracoderms, also unlike living jawless fishes, may have had paired fins. We should note, however, that the ostracoderms represent an assemblage of heavily armored extinct jawless fishes that may not form a natural evolutionary group. Fossils of the genus Hycuchthus from China, with an age of about 530 million years, show many typical vertebrate characteristics including paired eyes, auditory capsules, and rudimentary vertebrae. Class Myxinae. Hagfishes. The class Myxinae includes at least 70 species of hagfishes, eel-like scavengers that live on the ocean floor and feed on living or dead invertebrates, fishes, and marine mammals, figure 29.9. Although they are almost completely blind, sensory barbels around the mouth help them locate food by smell and touch. They feed using keratinized teeth on a movable cartilaginous plate in the mouth, which rasp pieces of flesh from their prey. These feeding structures allow the gills to be used exclusively for respiration, not for filter feeding as in the urochordates and cephalochordates. Hagfishes are entirely marine and are found in oceans around the world, except for the polar regions. Unique slime glands beneath the skin release a milky mucus, through surface pores, that upon contact with water becomes incredibly slippery, making the animal almost impossible to hold. This slippery mucus thus allows the hagfish to escape from the grip of predators. Hagfish can also twist their bodies into a knot, which provides additional leverage to feed. Sometimes hagfish enter the bodies of dead animals and eat carcasses from the inside out. Interestingly, they do not have a stomach. 29.2, Fishes 785 figure 29.9 Hagfish. Pacific hagfish are scavengers that live on the ocean floor. Credit. Linda Snook, NOAA, CBNMS. Hagfishes have a cartilaginous skull, as well as a fibrous and cartilaginous skeleton, but the major supportive structure is the notochord that runs the length of the body. In hagfishes, the notochord is not replaced by the vertebral column, as it is in true vertebrates, and thus they may, morphologically, represent a sister group to the true vertebrates, making them the most basal clade among the skull-bearing chordates. Class Petromyzontida. Lampreys. The class Petromyzontida includes approximately 40 species of lampreys, which are superficially similar to hagfishes in size and shape. However, lampreys possess extrinsic eye muscles, at least 2 semicircular canals, and a true cerebellum, as well as simple vertebral elements, called arqualia, cartilaginous structures arranged above the notochord. These features are also shared with the gnathostomes, vertebrates with jawed mouths and paired appendages, see below. Lampreys also have a dorsal tubular nerve cord with a well-differentiated brain, a small cerebellum, and 10 pairs of nerves. The classification of lampreys is still debated, but they clearly represent one of the oldest divergences of the vertebrate lineage. Lampreys lack paired appendages, as do the hagfishes, although they have 1 or 2 fleshy dorsal fins. As adults, lampreys are characterized by a rasping tongue within a toothed, funnel-like sucking mouth. Many species have a parasitic stage of their life cycle during which they are fish ectoparasites. Some call them predators because they attack and eventually fall off, figure 29.10. Figure 29.10 lamprey. These parasitic sea lampreys, Petromyzon marinus, attach by suction to their lake trout host, and use their rough tongues to rasp away flesh in order to feed on the trout's blood. Credit. USGS. Lampreys live primarily in coastal and freshwater environments, and have a worldwide distribution, except for the tropics and polar regions. Some species are marine, but all species spawn in fresh water. Interestingly, northern lampreys in the family Petromyzontidae have the highest number of chromosomes, 164 to 174, among the vertebrates. Eggs are fertilized externally, and the larvae, called amocetes, differ greatly from the adult form, closely resembling the adult cephalochordate Amphioxus. After spending 3 to 15 years as suspension feeders in rivers and streams, they attain sexual maturity. Shortly afterward, the adults swim upstream, reproduce, and die within days. 786 Chapter 29, Vertebrates access for free at OpenStax.org. Gnathostomes. Jawed fishes. Gnathostomes, or jaw mouths, are vertebrates that possess true jaws, a milestone in the evolution of the vertebrates. In fact, one of the most significant developments in early vertebrate evolution was the development of the jaw, a hinged structure attached to the cranium that allows an animal to grasp and tear its food. Jaws were probably derived from the first pair of gill arches supporting the gills of jawless fishes. Early gnathostomes also possessed 2 sets of paired fins, allowing the fishes to maneuver accurately. Pectoral fins are typically located on the anterior body, and pelvic fins on the posterior. Evolution of the jaw and paired fins permitted gnathostomes to expand their food options from the scavenging and suspension feeding of jawless fishes to active predation. The ability of gnathostomes to exploit new nutrient sources probably contributed to their replacing most jawless fishes during the Devonian period. 2 early groups of gnathostomes were the acanthodians and placoderms, figure 29.11, which arose in the late Silurian period and are now extinct. Most modern fishes are gnathostomes that belong to the clades Chondrichthyes and Osteichthyes, which include the class Actinotertigii and class Sarcoterygii. Figure 29.11a placoderm. Dunkleosteus was an enormous placoderm from the Devonian period, 380 to 360 million years ago. It measured up to 10 meters in length and weighed up to 3.6 tons. Its head and neck were armored with heavy bony plates. Although Dunkleosteus had no true teeth, the edge of the jaw was armed with sharp bony blades. Credit. Nobu Tamura. Class Chondrichthyes. Cartilaginous fishes. The class Chondrichthyes, about 1,000 species, is a morphologically diverse clade, consisting of subclass Elasmobranchii, sharks, figure 29.12, rays, and skates, together with the obscure and critically endangered sawfishes, and a few dozen species of fishes called chimeras, or ghost sharks, in the subclass Holocephala. Chondrichthyes are jawed fishes that possess paired fins and a skeleton made of cartilage. This clade arose approximately 370 million years ago in the early or middle Devonian. They are thought to be descended from the placoderms, which had endoskeletons made of bone. Thus, the lighter cartilaginous skeleton of Chondrichthyes is a secondarily derived evolutionary development. Parts of shark skeleton are strengthened by granules of calcium carbonate, but this is not the same as bone. Most cartilaginous fishes live in marine habitats, with a few species living in freshwater for a part or all of their lives. Most sharks are carnivores that feed on live prey, either swallowing it whole or using their jaws and teeth to tear it into smaller pieces. Sharks have abrasive skin covered with tooth-like scales called placoid scales. Shark teeth probably evolved from rows of these scales lining the mouth. A few species of sharks and rays, like the enormous whale shark, figure 29.13, are suspension feeders that feed on plankton. The sawfishes have an extended rostrum that looks like a double-edged saw. The rostrum is covered with electrosensitive pores that allow the sawfish to detect slight movements of prey hiding in the muddy seafloor. The teeth in the rostrum are actually modified tooth-like structures called denticles, similar to scales. 29.2. Fishes. 787 figure 29.12 shark. Hammerhead sharks tend to school during the day and hunt prey at night. Credit. Masashi Sugawara. Sharks have well-developed sense organs that aid them in locating prey, including a keen sense of smell and the ability to detect electromagnetic fields. Electroreceptors called ampullae of Lorenzini allow sharks to detect the electromagnetic fields that are produced by all living things, including their prey. Electroreception has only been observed in aquatic or amphibious animals, and sharks have perhaps the most sensitive electroreceptors of any animal. Sharks, together with most fishes and aquatic and larval amphibians, also have a row of sensory structures called the lateral line, which is used to detect movement and vibration in the surrounding water, and is often considered to be functionally similar to the sense of hearing in terrestrial vertebrates. The lateral line is visible as a darker stripe that runs along the length of a fish's body. Sharks have no mechanism for maintaining neutral buoyancy and must swim continuously to stay suspended in the water. Some must also swim in order to ventilate their gills, but others have muscular pumps in their mouths to keep water flowing over the gills. Figure 29.13 Whale shark in the Georgia Aquarium. Whale sharks are filter feeders and can grow to be over 10 meters long. Whale sharks, like most other sharks, are ovoviviparous. Credit. Modified from Zach Wolf, own work. CC by SA 2.5. http://creativecommons.org/licenses/by SA 2.5. http://openstax.org/l/ccsa. Via Wikimedia Commons, sharks reproduce sexually, and eggs are fertilized internally. Most species are ovoviviparous. The fertilized egg is retained in the oviduct of the mother's body and the embryo is nourished by the egg yolk. The eggs hatch in the uterus, and young are born alive and fully functional. Some species of sharks are oviparous. They lay eggs that hatch outside of the mother's body. Embryos are protected by a shark egg case or mermaid's purse, figure 29.14, that has the consistency of leather. The shark egg case has tentacles that snag in seaweed and give the newborn shark cover. A few species of sharks, e.g. tiger sharks and hammerheads, are viviparous. The yolk sac that initially contains the egg yolk and transfers its nutrients to the growing embryo becomes attached to the oviduct of the female, and nutrients are transferred directly from the mother to the growing embryo. In both viviparous and ovoviviparous sharks, gas exchange uses this yolk sac transport. 788 Chapter 29, Vertebrates access for free at openstax.org. Figure 29.14 Shark egg cases. Shark embryos are clearly visible through these transparent egg cases. The round structure is the yolk that nourishes the growing embryo. Credit. Jecpicarices, in general, the chondrichthyes have a fusiform or dorsoventrally flattened body, a heterocercal caudal fin or tail, unequally sized fin lobes, with the tail vertebrae extending into the larger upper lobe, paired pectoral and pelvic fins. In males these may be modified as claspers, exposed gill slits, elasmobranch, and an intestine with a spiral valve that condenses the length of the intestine. They also have 3 pairs of semicircular canals, and excellent senses of smell, vibration, vision, and electroreception. A very large lobed liver produces squalene oil, a lightweight biochemical precursor to steroids, that serves to aid in buoyancy, because with a specific gravity of 0.855, it is lighter than that of water. Rays and skates comprise more than 500 species. They are closely related to sharks but can be distinguished from sharks by their flattened bodies, pectoral fins that are enlarged and fused to the head, and gill slits on their ventral surface, figure 29.15. Like sharks, rays and skates have a cartilaginous skeleton. Most species are marine and live on the seafloor, with nearly a worldwide distribution. Unlike the stereotypical sharks and rays, the holocephala, chimeras or ratfish, have a difficircle tail, equally sized fin lobes, with the tail vertebrae located between them, lack scales, lost secondarily in evolution, and have teeth modified as grinding plates that are used to feed on mollusks and other invertebrates, figure 29.15b. Unlike sharks with elasmobranch or naked gills, chimeras have 4 pairs of gills covered by an operculum. Many species have a pearly iridescence and are extremely pretty. Figure 29.15 Cartilaginous fish. A. Stingray. This stingray blends into the sandy bottom of the ocean floor. A spotted ratfish. B. Hydrologus caliae credita, Salm 1-inch, flicker. Credit. A. Salm 1-inch, flicker. B. Linda Snook, MBNMS. Public domain, via Wikimedia Commons. Osteichthyes. Bony fishes, members of the clade Osteichthyes, also called bony fishes, are characterized by a bony skeleton. The vast majority of present-day fishes belong to this group, which consists of approximately 30,000 species, making it the largest class of vertebrates in existence today. Nearly all bony fishes have an ossified skeleton with specialized bone cells, osteocytes, that produce and maintain a calcium phosphate matrix. This characteristic has been reversed only in a few groups of Osteichthyes, such as sturgeons and paddlefish, 29.2, fishes 789, which have primarily cartilaginous skeletons. The skin of bony fishes is often covered by overlapping scales, and glands in the skin secrete mucus that reduces drag when swimming and aids the fish in osmoregulation. Like sharks, bony fishes have a lateral line system that detects vibrations in water. All bony fishes use gills to breathe. Water is drawn over gills that are located in chambers covered and ventilated by a protective, muscular flap called the operculum. Many bony fishes also have a swim bladder, a gas-filled organ derived as a pouch from the gut. The swim bladder helps to control the buoyancy of the fish. In most bony fish, the gases of the swim bladder are exchanged directly with the blood. The swim bladder is believed to be homologous to the lungs of lungfish and the lungs of land vertebrates. Bony fishes are further divided into two extant clades: class Actinopterygii, ray-finned fishes, and class Sarcopterygii, lobe-finned fishes. Actinopterygii, figure 29.16a, the ray-finned fishes, include many familiar fishes, tuna, bass, trout, and salmon among others, and represent about half of all vertebrate species. Ray-finned fishes are named for the fan of slender bones that supports their fins. In contrast, the fins of Sarcopterygii, figure 29.16b, are fleshy and lobed, supported by bones that are similar in type and arrangement to the bones in the limbs of early tetrapods. The few extant members of this clade include several species of lungfishes and the less familiar coelacanths, which were thought to be extinct until living specimens were discovered between Africa and Madagascar. Currently, two species of coelacanths have been described. Figure 29.16 Osteichthyes. The sockeye salmon and b. coelacanth are both bony fishes of the Osteichthyes clade. The coelacanth, sometimes called a lobe-finned fish, was thought to have gone extinct in the late Cretaceous period, 100 million years ago, until one was discovered in 1938 near the Comoros Islands between Africa and Madagascar. Credit A. Modification of work by Timothy Knepp, USFWS. Credit B. Modification of work by Robbie Cotta. 29.3 Amphibians. By the end of this section, you will be able to do the following. Describe the important difference between the life cycle of amphibians and the life cycles of other vertebrates. Distinguish between the characteristics of Urodela, Anura, and Apoda. Describe the evolutionary history of amphibians. Amphibians are vertebrate tetrapods, 4 limbs, and include frogs, salamanders, and caecilians. The term amphibian loosely translates from the Greek as dual life, which is a reference to the metamorphosis that many frogs and salamanders undergo and the unique mix of aquatic and terrestrial phases that are required in their life cycle. In fact, they cannot stray far from water because their reproduction is intimately tied to aqueous environments. Amphibians evolved during the Devonian period and were the earliest terrestrial tetrapods. They represent an evolutionary transition from water to land that occurred over many millions of years. Thus, the amphibia are the only living true vertebrates that have made a transition from water to land in both their ontogeny, life development, and phylogeny, evolution. They have not changed much in morphology over the past 350 million years. Link to learning. Watch this series of 5 Animal Planet videos on tetrapod evolution. 1. The evolution from fish to earliest tetrapod. Click to view content, https://www.openstax.org/l/tetrapod_evol1. 2. Fish to earliest tetrapod. Click to view content, https://www.openstax.org/l/tetrapod_evol2. 3. The discovery of coelacanth and Acanthostega fossils. 790. Chapter 29. Vertebrates. Access for free at openstax.org. Click to view content, https://www.openstax.org/l/tetrapod_evol3 4. The number of fingers on legs. Click to view content, https://www.openstax.org/l/tetrapod_evol4 5. Reconstructing the environment of early tetrapods. Click to view content, https://www.openstax.org/l/tetrapod_evol5 Characteristics of amphibians as tetrapods. Most amphibians are characterized by 4 well-developed limbs. In some species of salamanders, hind limbs are reduced or absent, but all caecilians are, secondarily, limbless. An important characteristic of extant amphibians is a moist, permeable skin that is achieved via mucus glands. Most water is taken in across the skin rather than by drinking. The skin is also one of 3 respiratory surfaces used by amphibians. The other 2 are the lungs and the buccal, mouth, cavity. Air is taken first into the mouth through the nostrils, and then pushed by positive pressure into the lungs by elevating the throat and closing the nostrils. All extant adult amphibians are carnivorous, and some terrestrial amphibians have a sticky tongue used to capture prey. Amphibians also have multiple small teeth at the edge of the jaws. In salamanders and caecilians, teeth are present in both jaws, sometimes in multiple rows. In frogs and toads, teeth are seen only in the upper jaw. Additional teeth, called vomerine teeth, may be found in the roof of the mouth. Amphibian teeth are pedicellate, which means that the root and crown are calcified, separated by a zone of non-calcified tissue. Amphibians have image-forming eyes and color vision. Ears are best developed in frogs and toads, which vocalize to communicate. Frogs use separate regions of the inner ear for detecting higher and lower sounds. The papilla amphibiorum, which is sensitive to frequencies below 10,000 Hz and unique to amphibians, and the papilla basilaris, which is sensitive to higher frequencies, including mating calls, transmitted from the eardrum through the stapes bone. Amphibians also have an extra bone in the ear, the operculum, which transmits low-frequency vibrations from the forelimbs and shoulders to the inner ear, and may be used for the detection of seismic signals. Evolution of amphibians. The fossil record provides evidence of the first tetrapods, now extinct amphibian species dating to nearly 400 million years ago. Evolution of tetrapods from lobe-finned freshwater fishes, similar to coelacanths and lungfish, represented a significant change in body plan from one suited to organisms that respired and swam in water, to organisms that breathed air and moved onto land. These changes occurred over a span of 50 million years during the Devonian period. Aquatic tetrapods of the Devonian period include Ichthyostega and Acanthostega. Both were aquatic, and may have had both gills and lungs. They also had 4 limbs, with the skeletal structure of limbs found in present-day tetrapods, including amphibians. However, the limbs could not be pulled in under the body and would not have supported their bodies well out of water. They probably lived in shallow freshwater environments, and may have taken brief terrestrial excursions, much like walking catfish do today in Florida. In Ichthyostega, the forelimbs were more developed than the hind limbs, so it might have dragged itself along when it ventured onto land. What preceded Acanthostega and Ichthyostega? In 2006, researchers published news of their discovery of a fossil of a tetrapod-like fish, Tiktaalik rosei, which seems to be a morphologically intermediate form between sarcopterygian fishes having feet like fins and early tetrapods having true limbs. Figure 29.17. Tiktaalik likely lived in a shallow water environment about 375 million years ago. 2. Tiktaalik also had gills and lungs, but the loss of some gill elements gave it a neck, which would have allowed its head to move sideways for feeding. The eyes were on top of the head. It had fins, but the attachment of the fin bones to the shoulder suggested they might be weight-bearing. Tiktaalik preceded Acanthostega and Ichthyostega, with their 4 limbs, by about 10 million years and is considered to be a true intermediate clade between fish and amphibians. 2. Deichler, E.B., Shubin, N.H., and Jenkins, F.J., A Devonian Tetrapod-like Fish and the Evolution of the Tetrapod Body Plan, Nature 440, 2006, 757–763, doi. 10.1038, Nature 04639. http://www.nature.com/nature/journal/v440/n7085/ab/nature04639.html http://openstax.org/l/tetrapod 29.3 Amphibians 791 Figure 29.17 Tiktaalik. The recent fossil discovery of Tiktaalik rosei suggests evidence for an animal intermediate to finned fish and legged tetrapods, sometimes called a fishapod. Credit: Zina Duretsky, National Science Foundation. The early tetrapods that moved onto land had access to new nutrient sources and relatively few predators. This led to the widespread distribution of tetrapods during the early Carboniferous period, a period sometimes called the Age of the Amphibians. Modern amphibians. Amphibia comprises an estimated 6,770 extant species that inhabit tropical and temperate regions around the world. All living species are classified in the subclass Lysamphibia, smooth amphibian, which is divided into three clades: Urodela, tailed, the salamanders; Anura, tailless, the frogs; and Apoda, legless ones, the caecilians. Urodela. Salamanders are amphibians that belong to the order Urodela, or Caudata. These animals are probably the most similar to ancestral amphibians. Living salamanders, figure 29.18, include approximately 620 species, some of which are aquatic, others terrestrial, and some that live on land only as adults. Most adult salamanders have a generalized tetrapod body plan with 4 limbs and a tail. The placement of their legs makes it difficult to lift their bodies off the ground, and they move by bending their bodies from side to side, called lateral undulation, in a fish-like manner while walking, their arms and legs fore and aft. It is thought that their gait is similar to that used by early tetrapods. The majority of salamanders are lungless, and respiration occurs through the skin or through external gills in aquatic species. Some terrestrial salamanders have primitive lungs. A few species have both gills and lungs. The giant Japanese salamander, the largest living amphibian, has additional folds in its skin that enlarge its respiratory surface. Most salamanders reproduce using an unusual process of internal fertilization of the eggs. Mating between salamanders typically involves an elaborate and often prolonged courtship. Such a courtship ends in the deposition of sperm by the males in a packet called a spermatophore, which is subsequently picked up by the female, thus ultimately fertilization is internal. All salamanders except one, the fire salamander, are oviparous. Aquatic salamanders lay their eggs in water, where they develop into legless larvae called efts. Terrestrial salamanders lay their eggs in damp nests, where the eggs are guarded by their mothers. These embryos go through the larval stage and complete metamorphosis before hatching into tiny adult forms. One aquatic salamander, the Mexican axolotl, never leaves the larval stage, becoming sexually mature without metamorphosis. 792 Chapter 29, Vertebrates access for free at OpenStax.org. Figure 29.18 Salamander. Most salamanders have legs and a tail, but respiration varies among species. Credit: Valentina Storti. Link to Learning View River Monsters. Fish with Arms and Hands. http://openstax.org/l/river_monster to see a video about an unusually large salamander species. Anura. Frogs, figure 29.19, are amphibians that belong to the order Anura or Salientia, jumpers. Anurans are among the most diverse groups of vertebrates, with approximately 5,965 species that occur on all of the continents except Antarctica. Anurans, ranging from the minute New Guinea frog at 7 mm to the huge Goliath frog at 32 cm from tropical Africa, have a body plan that is more specialized for movement. Adult frogs use their hind limbs and their arrow-like endoskeleton to jump accurately to capture prey on land. Tree frogs have hands adapted for grasping branches as they climb. In tropical areas, flying frogs can glide from perch to perch on the extended webs of their feet. Frogs have a number of modifications that allow them to avoid predators, including skin that acts as camouflage. Many species of frogs and salamanders also release defensive chemicals that are poisonous to predators from glands in the skin. Frogs with more toxic skins have bright warning, aposematic, coloration. Figure 29.19 Tree frog. The Australian green tree frog is a nocturnal predator that lives in the canopies of trees near a water source. Frog eggs are fertilized externally, and like other amphibians, frogs generally lay their eggs in moist environments. Although amphibian eggs are protected by a thick jelly layer, they would still dehydrate quickly in a dry environment. Frogs demonstrate a great diversity of parental behaviors, with some species laying many eggs and exhibiting little parental care, to species that carry eggs and tadpoles on their hind legs or embedded in their backs. The males of Darwin's frog carry tadpoles in their vocal sac. Many tree frogs lay their eggs off the ground in a folded leaf located over water so that the tadpoles can drop into the water as they hatch. The life cycle of most frogs, as other amphibians, consists of two distinct stages: the larval stage followed by metamorphosis to an adult stage. However, the eggs of frogs in the genus Eleutherodactylus develop directly into little froglets, guarded by a parent. The larval stage of a frog, the tadpole, is often a filter-feeding herbivore. Tadpoles usually have gills, a lateral line system, 29.3, amphibians 793 long-finned tails, and lack limbs. At the end of the tadpole stage, frogs undergo metamorphosis into the adult form, figure 29.20. During this stage, the gills, tail, and lateral line system disappear, and four limbs develop. The jaws become larger and are suited for carnivorous feeding, and the digestive system transforms into the typical short gut of a predator. An eardrum and air-breathing lungs also develop. These changes during metamorphosis allow the larva to move onto land in the adult stage. Figure 29.20 Amphibian metamorphosis. A juvenile frog metamorphoses into a frog. Here, the frog has started to develop limbs, but its tadpole tail is still evident. Apoda. Caecilians. An estimated 185 species comprise the caecilians, a group of amphibians that belong to the order Apoda. They have no limbs, although they evolved from a legged vertebrate ancestor. The complete lack of limbs makes them resemble earthworms. This resemblance is enhanced by folds of skin that look like the segments of an earthworm. However, unlike earthworms, they have teeth in both jaws, and feed on a variety of small organisms found in soil, including earthworms. Caecilians are adapted for a burrowing or aquatic lifestyle, and they are nearly blind, with their tiny eyes sometimes covered by skin. Although they have a single lung, they also depend on cutaneous respiration. These animals are found in the tropics of South America, Africa, and southern Asia. In the caecilians, the only amphibians in which the males have copulatory structures, fertilization is internal. Some caecilians are oviparous, but most bear live young. In these cases, the females help nourish their young with tissue from their oviduct before birth and from their skin after birth. Evolution connection. The Paleozoic era and the evolution of vertebrates. When the vertebrates arose during the Paleozoic era, 542 to 251 Maya, the climate and geography of Earth was vastly different. The distribution of landmasses on Earth were also very different from that of today. Near the equator were two large supercontinents, Laurentia and Gondwana, which included most of today's continents, but in a radically different configuration, figure 29.21. At this time, sea levels were very high, probably at a level that hasn't been reached since. As the Paleozoic progressed, glaciations created a cool global climate, but conditions warmed near the end of the first half of the Paleozoic. During the latter half of the Paleozoic, the landmasses began moving together, with the initial formation of a large northern block called Laurasia, which contained parts of what is now North America, along with Greenland, parts of Europe, and Siberia. Eventually, a single supercontinent, called Pangaea, was formed, starting in the latter third of the Paleozoic. Glaciations then began to affect Pangaea's climate and the distribution of vertebrate life. 794 Chapter 29, Vertebrates access for free at OpenStax.org. Figure 29.21 Paleozoic continents. During the Paleozoic era, around 550 million years ago, the continent Gondwana formed. Both Gondwana and the continent Laurentia were located near the equator. During the early Paleozoic, the amount of carbon dioxide in the atmosphere was much greater than it is today. This may have begun to change later, as land plants became more common. As the roots of land plants began to infiltrate rock and soil began to form, carbon dioxide was drawn out of the atmosphere and became trapped in the rock. This reduced the levels of carbon dioxide and increased the levels of oxygen in the atmosphere, so that by the end of the Paleozoic, atmospheric conditions were similar to those of today. As plants became more common through the latter half of the Paleozoic, microclimates began to emerge and ecosystems began to change. As plants and ecosystems continued to grow and become more complex, vertebrates moved from the water to land. The presence of shoreline vegetation may have contributed to the movement of vertebrates onto land. One hypothesis suggests that the fins of aquatic vertebrates were used to maneuver through this vegetation, providing a precursor to the movement of fins on land and the further development of limbs. The late Paleozoic was a time of diversification of vertebrates, as amniotes emerged and became two different lines that gave rise, on one hand, to synapsids and mammals, and, on the other hand, to the codonts, reptiles, dinosaurs, and birds. Many marine vertebrates became extinct near the end of the Devonian period, which ended about 360 million years ago, and both marine and terrestrial vertebrates were decimated by a mass extinction in the early Permian period about 250 million years ago. Link to Learning View Earth's Paleogeography, Continental Movements Through Time, http://openstax.org/l/paleogeography to see changes in Earth as life evolved. 29.4 Reptiles. By the end of this section, you will be able to do the following. Describe the main characteristics of amniotes. Explain the difference between anapsids, synapsids, and diapsids, and give an example of each. Identify the characteristics of reptiles. Discuss the evolution of reptiles. The reptiles, including dinosaurs and birds, are distinguished from amphibians by their terrestrially adapted egg, which is supported by four extraembryonic membranes: the yolk sac, the amnion, the chorion, and the allantois, figure 29.22. The chorion and amnion develop from folds in the body wall, and the yolk sac and allantois are extensions of the midgut and hindgut 29.4, reptiles 795 respectively. The amnion forms a fluid-filled cavity that provides the embryo with its own internal aquatic environment. The evolution of the extraembryonic membranes led to less dependence on water for development and thus allowed the amniotes to branch out into drier environments. In addition to these membranes, the eggs of birds, reptiles, and a few mammals have shells. An amniote embryo was then enclosed in the amnion, which was in turn encased in an extraembryonic coelom contained within the chorion. Between the shell and the chorion was the albumen of the egg, which provided additional fluid and cushioning. This was a significant development that further distinguishes the amniotes from amphibians, which were and continue to be restricted to moist environments due to their shell-less eggs. Although the shells of various reptilian amniotic species vary significantly, they all permit the retention of water and nutrients for the developing embryo. The egg shells of bird, avian reptiles, are hardened with calcium carbonate, making them rigid, but fragile. The shells of most non-avian reptile eggs, such as turtles, are leathery and require a moist environment. Most mammals do not lay eggs, except for monotremes such as the echidnas and platypuses. Instead, the embryo grows within the mother's body, with the placenta derived from two of the extraembryonic membranes. Characteristics of amniotes. The amniotic egg is the key characteristic of amniotes. In amniotes that lay eggs, the shell of the egg provides protection for the developing embryo while being permeable enough to allow for the exchange of carbon dioxide and oxygen. The albumen, or egg white, outside of the chorion provides the embryo with water and protein, whereas the fattier egg yolk contained in the yolk sac provides nutrients for the embryo, as is the case with the eggs of many other animals, such as amphibians. Here are the functions of the extraembryonic membranes. 1. Blood vessels in the yolk sac transport yolk nutrients to the circulatory system of the embryo. 2. The chorion facilitates exchange of oxygen and carbon dioxide between the embryo and the egg's external environment. 3. The allantois stores nitrogenous wastes produced by the embryo and also facilitates respiration. 4. The amnion protects the embryo from mechanical shock and supports hydration. In mammals, the yolk sac is very reduced, but the embryo is still cushioned and enclosed within the amnion. The placenta, which transports nutrients and functions in gas exchange and waste management, is derived from the chorion and allantois. Visual connection. Figure 29.22. An amniotic egg. The key features of an amniotic egg are shown. Which of the following statements about the parts of an egg are false? 1. The allantois stores nitrogenous waste and facilitates respiration. 2. The chorion facilitates gas exchange. 3. The yolk provides food for the growing embryo. 4. The amniotic cavity is filled with albumin. Additional derived characteristics of amniotes include a waterproof skin, accessory keratinized structures, and costal, rib. 796 Chapter 29. Vertebrates access for free at openstax.org. Ventilation of the lungs. Evolution of amniotes. The first amniotes evolved from tetrapod ancestors approximately 340 million years ago during the Carboniferous period. The early amniotes quickly diverged into two main lines, synapsids and sauropsids. Synapsids included the rhapsids, a clade from which mammals evolved. Sauropsids were further divided into anapsids and diapsids. Diapsids gave rise to the reptiles, including the dinosaurs and birds. The key differences between the synapsids, anapsids, and diapsids are the structures of the skull and the number of temporal fenestrae, windows, behind each eye. Figure 29.23. Temporal fenestrae are post-orbital openings in the skull that allow muscles to expand and lengthen. Anapsids have no temporal fenestrae, synapsids have one, fused ancestrally from two fenestrae, and diapsids have two, although many diapsids such as birds have highly modified diapsid skulls. Anapsids include extinct organisms and traditionally included turtles. However, more recent molecular and fossil evidence clearly shows that turtles arose within the diapsid line and secondarily lost the temporal fenestrae. Thus they appear to be anapsids because modern turtles do not have fenestrae in the temporal bones of the skull. The canonical diapsids include dinosaurs, birds, and all other extinct and living reptiles. Figure 29.23 Amniote skulls. Compare the skulls and temporal fenestrae of anapsids, synapsids, and diapsids. Anapsids have no openings, synapsids have one opening, and diapsids have two openings. The diapsids in turn diverged into two groups, the Archosauromorpha, ancient lizard form, and the Lepidosauromorpha, scaly lizard form, during the Mesozoic period. Figure 29.24. The lepidosaurs include modern lizards, snakes, and tuataras. The archosaurs include modern crocodiles and alligators, and the extinct ichthyosaurs, fish lizards, superficially resembling dolphins, pterosaurs, winged lizard, dinosaurs, terrible lizard, and birds. We should note that clade Dinosauria includes birds, which evolved from a branch of Maniraptoran theropod dinosaurs in the Mesozoic. The evolutionarily derived characteristics of amniotes include the amniotic egg and its four extraembryonic membranes, a thicker and more waterproof skin, and rib ventilation of the lungs. Ventilation is performed by drawing air into and out of the lungs by muscles such as the costal rib muscles and the diaphragm. 29.4. Reptiles. 797 Visual connection. Figure 29.24 Amniote phylogeny. This chart shows the evolution of amniotes. The placement of Testudines, turtles, is currently still debated. Question. Members of the order Testudines have an anapsid-like skull without obvious temporal fenestrae. However, molecular studies clearly indicate that turtles descended from a diapsid ancestor. Why might this be the case? In the past, the most common division of amniotes has been into the classes Mammalia, Reptilia, and Aves. However, both birds and mammals are descended from different amniote branches. The synapsids giving rise to the rhapsids and mammals, and the diapsids giving rise to the lepidosaurs and archosaurs. We will consider both the birds and the mammals as groups distinct from reptiles for the purpose of this discussion, with the understanding that this does not accurately reflect phylogenetic history and relationships. Characteristics of reptiles. Reptiles are tetrapods. Limbless reptiles, snakes and legless lizards, are classified as tetrapods because they are descended from four-limbed ancestors. Reptiles lay calcareous or leathery eggs enclosed in shells on land. Even aquatic reptiles return to the land to lay eggs. They usually reproduce sexually with internal fertilization. Some species display ovoviviparity, with the eggs remaining in the mother's body until they are ready to hatch. In ovoviviparous reptiles, most nutrients are supplied by the egg yolk, while the chorioallantois assists with respiration. Other species are viviparous, with the offspring born alive, with their development supported by a yolk sac placenta, a chorioallantoic placenta, or both. One of the key adaptations that permitted reptiles to live on land was the development of their scaly skin, containing the protein keratin and waxy lipids, which reduced water loss from the skin. A number of keratinous epidermal structures have emerged in the descendants of various reptilian lineages, and some have become defining characters for these lineages: scales, claws, nails, horns, feathers, and hair. Their occlusive skin means that reptiles cannot use their skin for respiration, like amphibians, and thus all amniotes breathe with lungs. All reptiles grow throughout their lives and regularly shed their skin, both to accommodate their growth and to rid themselves of ectoparasites. Snakes tend to shed the entire skin at one time, but other reptiles shed their skins in patches. Reptiles ventilate their lungs using various muscular mechanisms to produce negative pressure, low pressure, within the lungs that allows them to expand and draw in air. In snakes and lizards, the muscles of the spine and ribs are used to expand or contract the rib cage. Since walking or running interferes with this activity, the squamates cannot breathe effectively while 798 Chapter 29, Vertebrates access for free at openstax.org. Running. Some squamates can supplement rib movement with buccal pumping through the nose, with the mouth closed. In crocodilians, the lung chamber is expanded and contracted by moving the liver, which is attached to the pelvis. Turtles have a special problem with breathing, because their rib cage cannot expand. However, they can change the pressure around the lungs by pulling their limbs in and out of the shell, and by moving their internal organs. Some turtles also have a posterior respiratory sac that opens off the hindgut that aids in the diffusion of gases. Most reptiles are ectotherms, animals whose main source of body heat comes from the environment. However, some crocodilians maintain elevated thoracic temperatures and thus appear to be at least regional endotherms. This is in contrast to true endotherms, which use heat produced by metabolism and muscle contraction to regulate body temperature over a very narrow temperature range, and thus are properly referred to as homeotherms. Reptiles have behavioral adaptations to help regulate body temperature, such as basking in sunny places to warm up through the absorption of solar radiation, or finding shady spots or going underground to minimize the absorption of solar radiation, which allows them to cool down and prevent overheating. The advantage of ectothermy is that metabolic energy from food is not required to heat the body. Therefore, reptiles can survive on about 10% of the calories required by a similarly sized endotherm. In cold weather, some reptiles such as the garter snake brumate. Brumation is similar to hibernation in that the animal becomes less active and can go for long periods without eating, but differs from hibernation in that brumating reptiles are not asleep or living off fat reserves. Rather, their metabolism is slowed in response to cold temperatures, and the animal is very sluggish. Evolution of reptiles. Reptiles originated approximately 300 million years ago during the Carboniferous period. One of the oldest known amniotes is Cassinaria, which had both amphibian and reptilian characteristics. One of the earliest undisputed reptile fossils was Hylonomus, a lizard-like animal about 20 centimeters long. Soon after the first amniotes appeared, they diverged into three groups: synapsids, anapsids, and diapsids, during the Permian period. The Permian period also saw a second major divergence of diapsid reptiles into stem archosaurs, predecessors of the codonts, crocodilians, dinosaurs, and birds, and lepidosaurs, predecessors of snakes and lizards. These groups remained inconspicuous until the Triassic period, when the archosaurs became the dominant terrestrial group, possibly due to the extinction of large-bodied anapsids and synapsids during the Permian-Triassic extinction. About 250 million years ago, archosaurs radiated into the pterosaurs and both saurischian, lizard hip, and ornithischian, bird hip, dinosaurs, see below. Although they are sometimes mistakenly called dinosaurs, the pterosaurs were distinct from true dinosaurs, figure 29.25. Pterosaurs had a number of adaptations that allowed for flight, including hollow bones. Birds also exhibit hollow bones, a case of convergent evolution. Their wings were formed by membranes of skin that attached to the long, fourth finger of each arm and extended along the body to the legs. Figure 29.25. Pterosaurs. Pterosaurs, such as this Quetzalcoatlus, which existed from the late Triassic to the Cretaceous period, 230 to 65.5 million years ago, possessed wings but are not believed to have been capable of powered flight. Instead, they may have been able to soar after launching from cliffs. Credit: Mark Whitten, Darren Nash, Archosaurs. Dinosaurs, fearfully great lizard, include the Saurischia, lizard-hipped, with a simple, three-pronged pelvis, and 29.4. Reptiles 799 Ornithischia, bird-hipped, dinosaurs with a more complex pelvis, superficially similar to that of birds. However, it is a fact that birds evolved from the Saurischian, lizard-hipped, lineage, not the Ornithischian, bird-hipped, lineage. Dinosaurs and their theropod descendants, the birds, are remnants of what was formerly a hugely diverse group of reptiles, some of which like Argentinosaurus were nearly 40 meters, 130 feet, in length and weighed at least 80,000 kilograms, 88 tons. They were the largest land animals to have lived, challenging and perhaps exceeding the great blue whale in size, but probably not weight, which could be greater than 200 tons. Herrerasaurus, a bipedal dinosaur from Argentina, was one of the earliest dinosaurs that walked upright with the legs positioned directly below the pelvis, rather than splayed outward to the sides as in the crocodilians. The Ornithischia were all herbivores, and sometimes evolved into crazy shapes, such as ankylosaur, armored tanks, and horned dinosaurs such as Triceratops. Some, such as Parasaurolophus, lived in great herds and may have amplified their species-specific calls through elaborate crests on their heads. Both the Ornithischian and Saurischian dinosaurs provided parental care for their broods, just as crocodilians and birds do today. The end of the age of dinosaurs came about 65 million years ago, during the Mesozoic, coinciding with the impact of a large asteroid that produced the Chicxulub crater, in what is now the Yucatan Peninsula of Mexico. Besides the immediate environmental disasters associated with this asteroid impacting the Earth at about 45,000 miles per hour, the impact may also have helped generate an enormous series of volcanic eruptions that changed the distribution and abundance of plant life worldwide, as well as its climate. At the end of the Triassic, massive volcanic activity across North America, South America, Africa, and Southwest Europe ultimately would lead to the breakup of Pangaea and the opening of the Atlantic Ocean. The formerly incredibly diverse dinosaurs, save for the evolving birds, met their extinction during this time period. Archosaurs. Pterosaurs. More than 200 species of pterosaurs have been described, and in their day, beginning about 230 million years ago, they were the undisputed rulers of the Mesozoic skies for over 170 million years. Recent fossils suggest that hundreds of pterosaur species may have lived during any given period, dividing up the environment much like birds do today. Pterosaurs came in amazing sizes and shapes, ranging in size from that of a small songbird to that of the enormous Quetzalcoatlus northropi, which stood nearly 6 meters, 19 feet, high and had a wingspan of nearly 14 meters, 40 feet. This monstrous pterosaur, named after the Aztec god Quetzalcoatl, the feathered flying serpent that contributed largely to the creation of humankind, may have been the largest flying animal that ever evolved. Some male pterosaurs apparently had brightly colored crests that may have served in sexual displays. Some of these crests were much higher than the actual head. Pterosaurs had ultralight skeletons, with a pteroid bone, unique to pterosaurs, that strengthened the forewing membrane. Much of their wingspan was exaggerated by a greatly elongated fourth finger that supported perhaps half of the wing. It is tempting to relate to them in terms of bird characteristics, but in reality, their proportions were decidedly not like birds at all. For example, it is common to find specimens, such as Quetzalcoatlus, with a head and neck region that together was 3 to 4 times as large as the torso. In addition, unlike the feathered bird wing, the reptilian wing had a layer of muscles, connective tissue, and blood vessels, all reinforced with a webbing of fibrous cords. In contrast to the aerial pterosaurs, the dinosaurs were a diverse group of terrestrial reptiles with more than 1,000 species classified to date. Paleontologists continue to discover new species of dinosaurs. Some dinosaurs were quadrupeds, figure 29.26, others were bipeds. Some were carnivorous, whereas others were herbivorous. Dinosaurs laid eggs, and a number of nests containing fossilized eggs, with intact embryos, have been found. It is not known with certainty whether dinosaurs were homeotherms or facultative endotherms. However, given that modern birds are endothermic, the dinosaurs that were the immediate ancestors to birds likely were endothermic as well. Some fossil evidence exists for dinosaurian parental care, and comparative biology supports this hypothesis since the archosaur birds and crocodilians both display extensive parental care. 800 Chapter 29, Vertebrates access for free at OpenStax.org. Figure 29.26 Ornithischian and Saurischian dinosaurs. Edmontonia was an armored dinosaur that lived in the late Cretaceous period, 145.5 to 65.6 million years ago. Herrerasaurus and Eoraptor, B. were late Triassic Saurischian dinosaurs dating to about 230 million years ago. Credit. A. Mariana Ruiz Villarreal B. Zach Terrell from Plymouth, USA. Dino Origins. Dinosaurs dominated the Mesozoic era, which was known as the Age of Reptiles. The dominance of dinosaurs lasted until the end of the Cretaceous, the last period of the Mesozoic era. The Cretaceous Tertiary extinction resulted in the loss of most of the large-bodied animals of the Mesozoic era. Birds are the only living descendants of one of the major clades of theropod dinosaurs. Link to learning. Visit this site to see a video, http://openstax.org/l/k-t_extinction, discussing the hypothesis that an asteroid caused the Cretaceous-Triassic (KT) extinction. Modern reptiles. Class Reptilia includes many diverse species that are classified into 4 living clades. There are the 25 species of Crocodilia, 2 species of Sphenodontia, approximately 9,200 schemata species, and about 325 species of Testudines. Crocodilia. Crocodilia, small lizard, arose as a distinct lineage by the Middle Triassic. Extant species include alligators, crocodiles, gharials, and caimans. Crocodilians, figure 29.27, live throughout the tropics and subtropics of Africa, South America, southern Florida, Asia, and Australia. They are found in freshwater, saltwater, and brackish habitats, such as rivers and lakes, and spend most of their time in water. Crocodiles are descended from terrestrial reptiles and can still walk and run well on land. They often move on their bellies in a swimming motion, propelled by alternate movements of their legs. However, some species can lift their bodies off the ground, pulling their legs in under the body with their feet rotated to face forward. This mode of locomotion takes a lot of energy, and seems to be used primarily to clear ground obstacles. Amazingly, some crocodiles can also gallop, pushing off with their hind legs and moving their hind and forelegs alternately in pairs. Galloping crocodiles have been clocked at speeds over 17 km per hour and, over short distances, in an ambush situation, they can easily chase down most humans if they are taken by surprise. However, they are short-distance runners, not interested in a long chase, and most fit humans can probably outrun them in a sprint, assuming they respond quickly to the ambush. 29.4. Reptiles. 801 Figure 29.27a Crocodilian. Crocodilians, such as this Siamese crocodile, Crocodilus siamensis, provide parental care for their offspring. Credit. Keshav Mukund Candidae, Sphenodontia. Sphenodontia, wedge-tooth, arose in the early Mesozoic era, when they had a moderate radiation, but now are represented by only 2 living species, Sphenodon punctatus and Sphenodon guntheri, both found on offshore islands in New Zealand, figure 29.28. The common name, tuatara, comes from a Maori word describing the crest along its back. Tuataras have a primitive diapsid skull with biconcave vertebrae. They measure up to 80 centimeters and weigh about 1 kilogram. Although superficially similar to an iguanid lizard, several unique features of the skull and jaws clearly define them and distinguish this group from the schemata. They have no external ears. Tuataras briefly have a third, parietal, eye, with a lens, retina, and cornea, in the middle of the forehead. The eye is visible only in very young animals, it is soon covered with skin. Parietal eyes can sense light, but have limited color discrimination. Similar light-sensing structures are also seen in some other lizards. In their jaws, tuataras have 2 rows of teeth in the upper jaw that bracket a single row of teeth in the lower jaw. These teeth are actually projections from the jawbones, and are not replaced as they wear down. Figure 29.28a Tuatara. This tuatara from New Zealand may resemble a lizard but belongs to a distinct lineage, the Sphenodontidae family. Credit. Sid Mosdell, Schemata. The schemata, scaly or having scales, arose in the late Permian, and extant species include lizards and snakes. Both are found on all continents except Antarctica. Lizards and snakes are most closely related to tuataras, both groups having evolved from a lepidosaurian ancestor. Schemata is the largest extant clade of reptiles, Figure 29.29. 802 Chapter 29, Vertebrates access for free at OpenStax.org. Figure 29.29a Chameleon. This Jackson's chameleon, Trioceros jacksoni, blends in with its surroundings. Most lizards differ from snakes by having 4 limbs, although these have often been lost or significantly reduced in at least 60 lineages. Snakes lack eyelids and external ears, which are both present in lizards. There are about 6,000 species of lizards, ranging in size from tiny chameleons and geckos, some of which are only a few centimeters in length, to the Komodo dragon, which is about 3 meters in length. Some lizards are extravagantly decorated with spines, crests, and frills, and many are brightly colored. Some lizards, like chameleons, figure 29.29, can change their skin color by redistributing pigment within chromatophores in their skins. Chameleons change color both for camouflage and for social signaling. Lizards have multiple colored oil droplets in their retinal cells that give them a good range of color vision. Lizards, unlike snakes, can focus their eyes by changing the shape of the lens. The eyes of chameleons can move independently. Several species of lizards have a hidden parietal eye, similar to that in the tuatara. Both lizards and snakes use their tongues to sample the environment, and a pit in the roof of the mouth, Jacobson's organ, is used to evaluate the collected sample. Most lizards are carnivorous, but some large species, such as iguanas, are herbivores. Some predatory lizards are ambush predators, waiting quietly until their prey is close enough for a quick grab. Others are patient foragers, moving slowly through their environment to detect possible prey. Lizard tongues are long and sticky and can be extended at high speed for capturing insects or other small prey. Traditionally, the only venomous lizards are the Gila monster and the beaded lizard. However, venom glands have also been identified in several species of monitors and iguanids, but the venom is not injected directly and should probably be regarded as a toxin delivered with the bite. Specialized features of the jaw are related to adaptations for feeding that have evolved to feed on relatively large prey, even though some current species have reversed this trend. Snakes are thought to have descended from either burrowing or aquatic lizards over 100 million years ago, figure 29.30. They include about 3,600 species, ranging in size from 10 cm long thread snakes to 10 meter long pythons and anacondas. All snakes are legless, except for boids, e.g., boa constrictors, which have vestigial hind limbs in the form of pelvic spurs. Like Sicilian amphibians, the narrow bodies of most snakes have only a single functional lung. All snakes are carnivorous and eat small animals, birds, eggs, fish, and insects. 29.4. Reptiles 803 Figure 29.30 A non-venomous snake. The garter snake belongs to the genus Thamnophis, the most widely distributed reptile genus in North America. Credit: Steve Jurvetson. Most snakes have a skull that is very flexible, involving 8 rotational joints. They also differ from other squamates by having mandibles, lower jaws, without either bony or ligamentous attachment anteriorly. Having this connection via skin and muscle allows for great dynamic expansion of the gape and independent motion of the two sides, both advantages in swallowing big prey. Most snakes are non-venomous and simply swallow their prey alive, or subdue it by constriction before swallowing it. Venomous snakes use their venom both to kill or immobilize their prey, and to help digest it. Although snakes have no eyelids, their eyes are protected with a transparent scale. Their retinas have both rods and cones, and like many animals, they do not have receptor pigments for red light. Some species, however, can see in the ultraviolet, which allows them to track ultraviolet signals in rodent trails. Snakes adjust focus by moving their heads. They have lost both external and middle ears, although their inner ears are sensitive to ground vibrations. Snakes have a number of sensory structures that assist in tracking prey. In pit vipers, like rattlesnakes, a sensory pit between the eye and nostrils is sensitive to infrared, heat, emissions from warm-blooded prey. A row of similar pits is located on the upper lip of boids. As noted above, snakes also use Jacobson's organ for detecting olfactory signals. Testudines: the turtles, terrapins, and tortoises are members of the clade Testudines, having a shell. Figure 29.31, and are characterized by a bony or cartilaginous shell. The shell in turtles is not just an epidermal covering, but is incorporated into the skeletal system. The dorsal shell is called the carapace and includes the backbone and ribs. The ventral shell is called the plastron. Both shells are covered with keratinous plates or scutes, and the two shells are held together by a bridge. In some turtles, the plastron is hinged to allow the head and legs to be withdrawn under the shell. The two living groups of turtles, Pleurodira and Cryptodira, have significant anatomical differences and are most easily recognized by how they retract their necks. The more common Cryptodira retract their neck in a vertical S-curve. They appear to simply pull their head backward when retracting. Less common Pleurodira, side neck, retract their neck with a horizontal curve, basically folding their neck to the side. The testudines arose approximately 200 million years ago, predating crocodiles, lizards, and snakes. There are about 325 living species of turtles and tortoises. Like other reptiles, turtles are ectotherms. All turtles are oviparous, laying their eggs on land, although many species live in or near water. None exhibit parental care. Turtles range in size from the speckled padloper tortoise at 8 cm, 3.1 inches, to the leatherback sea turtle at 200 cm, over 6 feet. The term turtle is sometimes used to describe only those species of testudines that live in the sea, with the terms tortoise and terrapin used to refer to species that live on land and in fresh water, respectively. 804 Chapter 29, Vertebrates access for free at OpenStax.org. Figure 29.31a Tortoise. The African spurred tortoise, Geochelone sulcata, lives at the southern edge of the Sahara Desert. It is the third largest tortoise in the world. Credit. Jim Bowen. 29.5 Birds. By the end of this section, you will be able to do the following. Describe the evolutionary history of birds. Describe the derived characteristics in birds that facilitate flight with over 10,000 identified species. The birds are the most speciose of the land vertebrate classes. Abundant research has shown that birds are really an extant clade that evolved from Maniraptor and theropod dinosaurs about 150 million years ago. Thus, even though the most obvious characteristic that seems to set birds apart from other extant vertebrates is the presence of feathers, we now know that feathers probably appeared in the common ancestor of both Ornithischian and Saurischian lineages of dinosaurs. Feathers in these clades are also homologous to reptilian scales and mammalian hair, according to the most recent research. While the wings of vertebrates like bats function without feathers, birds rely on feathers and wings, along with other modifications of body structure and physiology, for flight, as we shall see. Characteristics of birds are endothermic, and more specifically, homeothermic, meaning that they usually maintain an elevated and constant body temperature, which is significantly above the average body temperature of most mammals. This is, in part, due to the fact that active flight, especially the hovering skills of birds such as hummingbirds, requires enormous amounts of energy, which in turn necessitates a high metabolic rate. Like mammals, which are also endothermic and homeothermic and covered with an insulating pelage, birds have several different types of feathers that together keep heat, infrared energy, within the core of the body, away from the surface where it can be lost by radiation and convection to the environment. Modern birds produce two main types of feathers: contour feathers and down feathers. Contour feathers have a number of parallel barbs that branch from a central shaft. The barbs in turn have microscopic branches called barbules that are linked together by minute hooks, making the vein of a feather a strong, flexible, and uninterrupted surface. In contrast, the barbules of down feathers do not interlock, making these feathers especially good for insulation, trapping air in spaces between the loose, interlocking barbules of adjacent feathers to decrease the rate of heat loss by convection and radiation. Certain parts of a bird's body are covered in down feathers, and the base of other feathers has a downy portion, whereas newly hatched birds are covered almost entirely in down, which serves as an excellent coat of insulation, increasing the thermal boundary layer between the skin and the outside environment. Feathers not only provide insulation, but also allow for flight, producing the lift and thrust necessary for flying birds to become and stay airborne. The feathers on a wing are flexible, so the feathers at the end of the wings separate as air moves over them, reducing the drag on the wing. Flight feathers are also asymmetrical and curved, so that air flowing over them generates lift. Two types of flight feathers are found on the wings: primary feathers and secondary feathers. Figure 29.32. Primary feathers are located at the tip of the wing and provide thrust as the bird moves its wings downward, using the pectoralis major muscles. 29.5. Birds' 805 secondary feathers are located closer to the body, in the forearm portion of the wing, and provide lift. In contrast to primary and secondary feathers, contour feathers are found on the body, where they help reduce form drag produced by wind resistance against the body during flight. They create a smooth, aerodynamic surface so that air moves swiftly over the bird's body, preventing turbulence and creating ideal aerodynamic conditions for efficient flight. Figure 29.32 Flight feathers. A. Primary feathers are located at the wingtip and provide thrust. Secondary feathers are located close to the body and provide lift. B. Primary and secondary feathers from a common buzzard, buteo buteo. Credit B. Mod. From S. Seifert https://commons.wikimedia.org/w/index.php?current equals 613813 http://openstax.org/l/buzzard_feathers closing parenthesis. Flapping of the entire wing occurs primarily through the actions of the chest muscles. Specifically, the contraction of the pectoralis major muscles moves the wings downward, downstroke, whereas contraction of the supracoracoideus muscles moves the wings upward, upstroke, via a tough tendon that passes over the coracoid bone and the top of the humerus. Both muscles are attached to the keel of the sternum, and these are the muscles that humans eat on holidays. This is why the back of the bird offers little meat. These muscles are highly developed in birds and account for a higher percentage of body mass than in most mammals. The flight muscles attach to a blade-shaped keel projecting ventrally from the sternum, like the keel of a boat. The sternum of birds is deeper than that of other vertebrates, which accommodates the large flight muscles. The flight muscles of birds who are active flyers are rich with oxygen-storing myoglobin. Another skeletal modification found in most birds is the fusion of the two clavicles, collarbones, forming the furcula or wishbone. The furcula is flexible enough to bend and provide support to the shoulder girdle during flapping. An important requirement for flight is a low body weight. As body weight increases, the muscle output required for flying increases. The largest living bird is the ostrich, and while it is much smaller than the largest mammals, it is secondarily flightless. For birds that do fly, reduction in body weight makes flight easier. Several modifications are found in birds to reduce body weight, including pneumatization of bones. Pneumatic bones, figure 29.33, are bones that are hollow, rather than filled with tissue. Cross struts of bone called trabeculae provide structural reinforcement. Pneumatic bones are not found in all birds, and they are more extensive in large birds than in small birds. Not all bones of the skeleton are pneumatic, although the skulls of almost all birds are. The jaw is also lightened by the replacement of heavy jawbones and teeth with a beak made of keratin, just as hair, scales, and feathers are. 806 Chapter 29, Vertebrates access for free at openstax.org. Figure 29.33 Pneumatic bone. Many birds have hollow, pneumatic bones, which make flight easier. Other modifications that reduce weight include the lack of a urinary bladder. Birds possess a cloaca, an external body cavity into which the intestinal, urinary, and genital orifices empty in reptiles, birds, and the monotreme mammals. The cloaca allows water to be reabsorbed from waste back into the bloodstream. Thus, uric acid is not eliminated as a liquid but is concentrated into urate salts, which are expelled along with fecal matter. In this way, water is not held in a urinary bladder, which would increase body weight. In addition, the females of most bird species only possess one functional, left, ovary rather than two, further reducing body mass. The respiratory system of birds is dramatically different from that of reptiles and mammals, and is well adapted for the high metabolic rate required for flight. To begin, the air spaces of pneumatic bone are sometimes connected to air sacs in the body cavity, which replace coulombic fluid and also lighten the body. These air sacs are also connected to the path of airflow through the bird's body, and function in respiration. Unlike mammalian lungs in which air flows in two directions, as it is breathed in and out, diluting the concentration of oxygen, airflow through bird lungs is unidirectional. Figure 29.34. Gas exchange occurs in air capillaries, or microscopic air passages within the lungs. The arrangement of air capillaries in the lungs creates a countercurrent exchange system with the pulmonary blood. In a countercurrent system, the air flows in one direction and the blood flows in the opposite direction, producing a favorable diffusion gradient and creating an efficient means of gas exchange. This very effective oxygen delivery system of birds supports their higher metabolic activity. In effect, ventilation is provided by the parabronchi, minimally expandable lungs, with thin air sacs located among the visceral organs and the skeleton. A syrinx, voice box, resides near the junction of the trachea and bronchi. The syrinx, however, is not homologous to the mammalian larynx, which resides within the upper part of the trachea. Figure 29.34 Airflow in bird lungs. Avian respiration is an efficient system of gas exchange with air flowing unidirectionally. A full ventilation cycle takes 2 breathing cycles. During the first inhalation, air passes from the trachea into posterior air sacs, then during the first exhalation into the lungs. The second inhalation moves the air in the lungs to the anterior air sacs, and the second exhalation moves the air in the anterior air sacs out of the body. Overall, each inhalation moves air into the air sacs, while each exhalation moves fresh air through the lungs and used air out of the body. The air sacs are connected to the hollow interior of bones. Credit. Modification of work by L. Shyamul. Beyond the unique characteristics discussed above, birds are also unusual vertebrates because of a number of other features. First, they typically have an elongate, very dinosaurian, S-shaped neck, but a short tail or pygostyle, produced from the fusion of the caudal vertebrae. Unlike mammals, birds have only one occipital condyle, allowing them extensive movement of the head and neck. They also have a very thin epidermis without sweat glands, and a specialized uropygial gland or sebaceous, preening gland, found at the dorsal base of the tail. This gland is an essential to preening, a virtually continuous activity, in most birds because it produces an oily substance that birds use to help waterproof their feathers as well as keep them flexible for flight. A 29.5, birds 807 number of birds, such as pigeons, parrots, hawks, and owls, lack a uropygial gland but have specialized feathers that disintegrate into a powdery down, which serves the same purpose as the oils of the uropygial gland. Like mammals, birds have 12 pairs of cranial nerves, and a very large cerebellum and optic lobes, but only a single bone in the middle ear called the columella, the stapes in mammals. They have a closed circulatory system with 2 atria and 2 ventricles, but rather than a left-bending, aortic arch like that of mammals, they have a right-bending, aortic arch, and nucleated red blood cells, unlike the enucleated red blood cells of mammals. All these unique and highly derived characteristics make birds one of the most conspicuous and successful groups of vertebrate animals, filling a range of ecological niches, and ranging in size from the tiny bee hummingbird of Cuba, about 2 grams, to the ostrich, about 140,000 grams. Their large brains, keen senses, and the abilities of many species to imitate vocalization and use tools make them some of the most intelligent vertebrates on Earth. Evolution of birds. Thanks to amazing new fossil discoveries in China, the evolutionary history of birds has become clearer, even though bird bones do not fossilize as well as those of other vertebrates. As we've seen earlier, birds are highly modified diapsids, but rather than having 2 fenestrations or openings in their skulls behind the eye, the skulls of modern birds are so specialized that it is difficult to see any trace of the original diapsid condition. Birds belong to a group of diapsids called the archosaurs, which includes 3 other groups: living crocodilians, pterosaurs, and dinosaurs. Overwhelming evidence shows that birds evolved within the clade Dinosauria, which is further subdivided into 2 groups, the Saurischia, lizard hips, and the Ornithischia, bird hips. Despite the names of these groups, it was not the bird-hipped dinosaurs that gave rise to modern birds. Rather, Saurischia diverged into 2 groups. One included the long-necked herbivorous dinosaurs, such as Apatosaurus. The second group, bipedal predators called theropods, gave rise to birds. This course of evolution is highlighted by numerous similarities between late, Maniraptoran, theropod fossils and birds, specifically in the structure of the hip and wrist bones, as well as the presence of the wishbone, formed by the fusion of the clavicles. The clade Neorniths includes the avian crown group, which comprises all living birds and the descendants from their most recent common Maniraptoran ancestor. One well-known and important fossil of an animal that appears intermediate between dinosaurs and birds is Archaeopteryx, figure 29.35, which is from the Jurassic period, 200 to 145 Maya. Archaeopteryx has characteristics of both Maniraptoran dinosaurs and modern birds. Some scientists propose classifying it as a bird, but others prefer to classify it as a dinosaur. Traits in skeletons of Archaeopteryx like those of a dinosaur included a jaw with teeth and a long bony tail. Like birds, it had feathers modified for flight, both on the forelimbs and on the tail, a trait associated only with birds among modern animals. Fossils of older feathered dinosaurs exist, but the feathers may not have had the characteristics of modern flight feathers. Figure 29.35 Archaeopteryx. A. Archaeopteryx lived in the late Jurassic period around 150 million years ago. It had cup-like the codont teeth like a dinosaur, but had, b. flight feathers like modern birds, which can be seen in this fossil. Note the claws on the wings, which are still found in a number of birds, such as the newborn chicks of the South American hoatzin. 808 Chapter 29. Vertebrates access for free at openstax.org. The evolution of flight in birds. There are two basic hypotheses that explain how flight may have evolved in birds. The arboreal, tree, hypothesis and the terrestrial, land, hypothesis. The arboreal hypothesis posits that tree-dwelling precursors to modern birds jumped from branch to branch using their feathers for gliding before becoming fully capable of flapping flight. In contrast to this, the terrestrial hypothesis holds that running, perhaps pursuing active prey such as small cursorial animals, was the stimulus for flight. In this scenario, wings could be used to capture prey and were pre-adapted for balance and flapping flight. Ostriches, which are large flightless birds, hold their wings out when they run, possibly for balance. However, this condition may represent a behavioral relic of the clade of flying birds that were their ancestors. It seems more likely that small feathered arboreal dinosaurs were capable of gliding and flapping from tree to tree and branch to branch, improving the chances of escaping enemies, finding mates, and obtaining prey such as flying insects. This early flight behavior would have also greatly increased the opportunity for species dispersal. Although we have a good understanding of how feathers and flight may have evolved, the question of how endothermy evolved in birds, and other lineages, remains unanswered. Feathers provide insulation, but this is only beneficial for thermoregulatory purposes if body heat is being produced internally. Similarly, internal heat production is only viable for the evolution of endothermy if insulation is present to retain that infrared energy. It has been suggested that one or the other, feathers or endothermy, evolved first in response to some other selective pressure, e.g., the ability to be active at night, provide camouflage, repel water, or serve as signals for mate selection. It seems probable that feathers and endothermy co-evolved together, the improvement and evolutionary advancement of feathers reinforcing the evolutionary advancement of endothermy, and so on. During the Cretaceous period, 145 to 66 Maya, a group known as the Enantiorniths was the dominant bird type, figure 29.36. Enantiorniths means opposite birds, which refers to the fact that certain bones of the shoulder are joined differently than the way the bones are joined in modern birds. Like Archaeopteryx, these birds retained teeth in their jaws, but did have a shortened tail, and at least some fossils have preserved fans of tail feathers. These birds formed an evolutionary lineage separate from that of modern birds, and they did not survive past the Cretaceous. Along with the Enantiorniths, however, another group of birds, the Ornithuri, birdtails, with a short, fused tail or pygostyle, emerged from the evolutionary line that includes modern birds. This clade was also present in the Cretaceous. After the extinction of Enantiorniths, the Ornithuri became the dominant birds, with a large and rapid radiation occurring after the extinction of the dinosaurs during the Cenozoic era, 66 Maya to the present. Molecular analysis based on very large datasets has produced our current understanding of the relationships among living birds. There are three major clades: the Paleognathi, the Galoncerae, and the Neoves. The Paleognathi, old jaw, or ratites, polyphyletic, are a group of flightless birds including ostriches, emus, rheas, and kiwis. The Galoncerae include pheasants, ducks, geese and swans. The Neoves, new birds, includes all other birds. The Neoves themselves have been distributed among 5 clades: 3 strosaurs, nightjars, swifts, and hummingbirds, columbaves, turacos, bustards, cuckoos, pigeons, and doves, gruiforms, cranes, acorlitorniths, diving birds, wading birds, and shorebirds, and anopanaves, a very large clade of land birds including hawks, owls, woodpeckers, parrots, falcons, crows, and songbirds. Despite the current classification scheme, it is important to understand that phylogenetic revisions, even for the extant birds, are still taking place. 3 Prum, R.O. et al. 2015. A Comprehensive Phylogeny of Birds, AVS, using targeted next-generation DNA sequencing. Nature 526, 569–573. http://dx.doi.org/10.1038/nature15697 http://openstax.org/l/bird_phylogeny 29.5 Birds 809 Figure 29.36 Enantiornithian bird. Shanweinau cooperorum was a species of enantiorniths that did not survive past the Cretaceous period. Credit. Nobu Tamura, Career Connection Veterinarians are concerned with diseases, disorders, and injuries in animals, primarily vertebrates. They treat pets, livestock, and animals in zoos and laboratories. Veterinarians often treat dogs and cats, but also take care of birds, reptiles, rabbits, and other animals that are kept as pets. Veterinarians that work with farms and ranches care for pigs, goats, cows, sheep, and horses. Veterinarians are required to complete a degree in veterinary medicine, which includes taking courses in comparative zoology, animal anatomy and physiology, microbiology, and pathology, among many other courses in chemistry, physics, and mathematics. Veterinarians are also trained to perform surgery on many different vertebrate species, which requires an understanding of the vastly different anatomies of various species. For example, the stomach of ruminants like cows has 4 compartments, versus 1 compartment for non-ruminants. As we have seen, birds also have unique anatomical adaptations that allow for flight, which requires additional training and care. Some veterinarians conduct research in academic settings, broadening our knowledge of animals and medical science. One area of research involves understanding the transmission of animal diseases to humans, called zoonotic diseases. For example, one area of great concern is the transmission of the avian flu virus to humans. One type of avian flu virus, H5N1, is a highly pathogenic strain that has been spreading in birds in Asia, Europe, Africa, and the Middle East. Although the virus does not cross over easily to humans, there have been cases of bird-to-human transmission. More research is needed to understand how this virus can cross the species barrier and how its spread can be prevented. 29.6 Mammals. By the end of this section, you will be able to do the following. Name and describe the distinguishing features of the 3 main groups of mammals. Describe the likely line of evolutionary descent that produced mammals. List some derived features that may have arisen in response to mammals' need for constant, high-level metabolism. Identify the major clades of eutherian mammals. Mammals, comprising about 5,200 species, are vertebrates that possess hair and mammary glands. Several other characteristics are distinctive to mammals, including certain features of the jaw, skeleton, integument, and internal anatomy. Modern mammals belong to 3 clades: monotremes, marsupials, and eutherians, or placental mammals. 810 Chapter 29, Vertebrates. Access for free at openstax.org. Characteristics of mammals. The presence of hair, composed of the protein keratin, is one of the most obvious characteristics of mammals. Although it is not very extensive or obvious on some species, such as whales, hair has many important functions for most mammals. Mammals are endothermic, and hair traps a boundary layer of air close to the body, retaining heat generated by metabolic activity. Along with insulation, hair can serve as a sensory mechanism via specialized hairs called vibrissae, better known as whiskers. Vibrissae attach to nerves that transmit information about tactile vibration produced by sound sensation, which is particularly useful to nocturnal or burrowing mammals. Hair can also provide protective coloration or be part of social signaling, such as when an animal's hair stands on end to warn enemies, or possibly to make the mammal look bigger to predators. Unlike the skin of birds, the integument, skin, of mammals, includes a number of different types of secretory glands. Sebaceous glands produce a lipid mixture called sebum that is secreted onto the hair and skin, providing water resistance and lubrication for hair. Sebaceous glands are located over most of the body. Eccrine glands produce sweat, or perspiration, which is mainly composed of water, but also contains metabolic waste products, and sometimes compounds with antibiotic activity. In most mammals, eccrine glands are limited to certain areas of the body, and some mammals do not possess them at all. However, in primates, especially humans, sweat glands are located over most of the body surface and figure prominently in regulating the body temperature through evaporative cooling. Apocrine glands, or scent glands, secrete substances that are used for chemical communication, such as in skunks. Mammary glands produce milk that is used to feed newborns. In both monotremes and eutherians, both males and females possess mammary glands, while in marsupials, mammary glands have been found only in some opossums. Mammary glands likely are modified sebaceous or eccrine glands, but their evolutionary origin is not entirely clear. The skeletal system of mammals possesses many unique features. The lower jaw of mammals consists of only one bone, the dentary, and the jaw hinge connects the dentary to the squamosal, flat, part of the temporal bone in the skull. The jaws of other vertebrates are composed of several bones, including the quadrate bone at the back of the skull and the articular bone at the back of the jaw, with the jaw connected between the quadrate and articular bones. In the ear of other vertebrates, vibrations are transmitted to the inner ear by a single bone, the stapes. In mammals, the quadrate and articular bones have moved into the middle ear, figure 29.37. The malleus is derived from the articular bone, whereas the incus originated from the quadrate bone. This arrangement of jaw and ear bones aids in distinguishing fossil mammals from fossils of other synapsids. Figure 29.37 Mammalian ear bones. Bones of the mammalian middle ear are modified from bones of the jaw and skull in reptiles. The stapes is found in other vertebrates, e.g., the columella of birds, whereas in mammals, the malleus and incus are derived from the articular and quadrate bones, respectively. Credit. NCI. The adductor muscles that close the jaw comprise two major muscles in mammals, the temporalis and the masseter. Working together, these muscles permit up and down and side-to-side movements of the jaw, making chewing possible, which is unique to mammals. Most mammals have heterodont teeth, meaning that they have different types and shapes of teeth, incisors, canines, premolars, and molars, rather than just one type and shape of tooth. Most mammals are also diphyodonts, meaning that they have two sets of teeth in their lifetime, deciduous or baby teeth, and permanent teeth. Most other vertebrates with teeth are polyphyodonts, that is, their teeth are replaced throughout their entire life. Mammals, like birds, possess a four-chambered heart. However, the hearts of birds and mammals are an example of convergent 29.6, mammals 811 evolution, since mammals clearly arose independently from different groups of tetrapod ancestors. Mammals also have a specialized group of cardiac cells, fibers, located in the walls of their right atrium called the sinoatrial node, or pacemaker, which determines the rate at which the heart beats. Mammalian erythrocytes, red blood cells, do not have nuclei, whereas the erythrocytes of other vertebrates are nucleated. The kidneys of mammals have a portion of the nephron called the loop of Henle or nephritic loop, which allows mammals to produce urine with a high concentration of solutes, higher than that of the blood. Mammals lack a renal portal system, which is a system of veins that moves blood from the hind or lower limbs and region of the tail to the kidneys. Renal portal systems are present in all other vertebrates except jawless fishes. A urinary bladder is present in all mammals. Unlike birds, the skulls of mammals have two occipital condyles, bones at the base of the skull that articulate with the first vertebra, as well as a secondary palate at the rear of the pharynx that helps to separate the pathway of swallowing from that of breathing. Turbinate bones, chonchi in humans, are located along the sides of the nasal cavity, and help warm and moisten air as it is inhaled. The pelvic bones are fused in mammals, and there are typically 7 cervical vertebrae, except for some edentates and manatees. Mammals have movable eyelids and fleshy external ears, pinnae, quite unlike the naked external auditory openings of birds. Mammals also have a muscular diaphragm that is lacking in birds. Mammalian brains also have certain characteristics that differ from the brains of other vertebrates. In some, but not all mammals, the cerebral cortex, the outermost part of the cerebrum, is highly convoluted and folded, allowing for a greater surface area than is possible with a smooth cortex. The optic lobes, located in the midbrain, are divided into two parts in mammals, while other vertebrates possess a single, undivided lobe. Eutherian mammals also possess a specialized structure, the corpus callosum, which links the two cerebral hemispheres together. The corpus callosum functions to integrate motor, sensory, and cognitive functions between the left and right cerebral cortexes. Evolution of mammals. Mammals are synapsids, meaning they have a single, ancestrally fused, postorbital opening in the skull. They are the only living synapsids, as earlier forms became extinct by the Jurassic period. The early non-mammalian synapsids can be divided into two groups, the pelycosaurs and the rhapsids. Within the rhapsids, a group called the cynodonts are thought to have been the ancestors of mammals, figure 29.38. Figure 29.38 Cynodont. Cynodonts, dogteeth, which first appeared in the late Permian period 260 million years ago, are thought to be the ancestors of modern mammals. Holes in the upper jaws of cynodonts suggest that they had whiskers, which might also indicate the presence of hair. Credit. Nobu Tamura. As with birds, a key characteristic of synapsids is endothermy, rather than the ectothermy seen in many other vertebrates, such as fish, amphibians, and most reptiles. The increased metabolic rate required to internally modify body temperature likely went hand in hand with changes to certain skeletal structures that improved food processing and ambulation. The later synapsids, which had more evolved characteristics unique to mammals, possessed cheeks for holding food and heterodont teeth, which are specialized for chewing, mechanically breaking down food to speed digestion, and releasing the energy needed to produce heat. Chewing also requires the ability to breathe at the same time, which is facilitated by the presence of a secondary palate, comprising the bony palate and the posterior continuation of the soft palate. The secondary palate separates the area of the mouth where chewing occurs from the area above where respiration occurs, allowing breathing to proceed uninterrupted while the animal is chewing. A secondary palate is not found in pelycosaurs but is present in cynodonts and mammals. The jawbone also shows changes from early synapsids to later ones. The zygomatic arch, or cheekbone, is present in mammals and advanced rhapsids such as cynodonts, but is not present in pelycosaurs. The presence of the zygomatic arch suggests the presence of 812 Chapter 29. Vertebrates access for free at openstax.org. Masseter muscles, which close the jaw and function in chewing. In the appendicular skeleton, the shoulder girdle of therian mammals is modified from that of other vertebrates in that it does not possess a procoracoid bone or an interclavicle, and the scapula is the dominant bone. Mammals evolved from the rhapsids in the late Triassic period, as the earliest known mammal fossils are from the early Jurassic period, some 205 million years ago. One group of transitional mammals was the Morgan eucodonts, small nocturnal insectivores. The jaws of Morgan eucodonts were transitional, with features of both reptilian and mammalian jaws, figure 29.39. Like modern mammals, the Morgan eucodonts had differentiated teeth and were diphyodonts. Mammals first began to diversify in the Mesozoic era, from the Jurassic to the Cretaceous periods. Even some small gliding mammals appear in the fossil record during this time period. However, most of the Jurassic mammals were extinct by the end of the Mesozoic. During the Cretaceous period, another radiation of mammals began and continued through the Cenozoic era, about 65 million years ago. Figure 29.39a Morganucodont. This Morganucodont megazotrodon, an extinct basal mammal, may have been nocturnal and insectivorous. Inset. Jaw of a Morganucodont, showing a double hinge, one between the dentary and squamosal and one between the articular, yellow, and quadrate, blue, bones. In living mammals, the articular and quadrate bones have been incorporated into the middle ear. Credit. By Nordelch, Megazostrodon Natural History Museum, Wikimedia Commons. Credit inset. Mod from Filcha. https://commons.wikimedia.org/w/index.php. Currid equals 3631949. http://openstax.org/l/jaw_joint closing parenthesis. Living mammals. There are three major groups of living mammals: monotremes, prototheria, marsupials, metatheria, and placental, eutheria, mammals. The eutherians and the marsupials together comprise a clade of therian mammals, with the monotremes forming a sister clade to both metatherians and eutherians. There are very few living species of monotremes, the platypus and 4 species of echidnas, or spiny anteaters. The leathery-beaked platypus belongs to the family Ornithorhynchidae, birdbeak, whereas echidnas belong to the family Tachyglossidae, sticky tongue. Figure 29.40. The platypus and 1 species of echidna are found in Australia, and the other species of echidna are found in New Guinea. Monotremes are unique among mammals because they lay eggs, rather than giving birth to live young. The shells of their eggs are not like the hard shells of birds, but have a leathery shell, similar to the shells of reptile eggs. Monotremes retain their eggs through about 2/3 of the developmental period, and then lay them in nests. A yolk sac placenta helps support development. The babies hatch in a fetal state and complete their development in the nest, nourished by milk secreted by mammary glands opening directly to the skin. Monotremes, except for young platypuses, do not have teeth. Body temperature in the 3 monotreme species is maintained at about 30 degrees Celsius, considerably lower than the average body temperature of marsupial and placental mammals, which are typically between 35 and 38 degrees Celsius. 29.6. Mammals 813. Figure 29.40. Egg-laying mammals. A. The platypus, a monotreme, possesses a leathery beak and lays eggs rather than giving birth to live young. B. The echidna is another monotreme, with long hairs modified into spines. Credit B. Modification of work by Barry Thomas. Over 2/3 of the approximately 330 living species of marsupials are found in Australia, with the rest, nearly all various types of opossum, found in the Americas, especially South America. Australian marsupials include the kangaroo, koala, bandicoot, Tasmanian devil, figure 29.41, and several other species. Like monotremes, the embryos of marsupials are nourished during a short gestational period, about a month in kangaroos, by a yolk sac placenta, but with no intervening eggshell. Some marsupial embryos can enter an embryonic diapause, and delay implantation, suspending development until implantation is completed. Marsupial young are also effectively fetal at birth. Most, but not all, species of marsupials possess a pouch in which the very premature young reside, receiving milk and continuing their development. In kangaroos, the young joeys continue to nurse for about a year and a half. Figure 29.41. A marsupial mammal. The Tasmanian devil is one of several marsupials native to Australia. Credit. Wayne McLean. Eutherians, placentals, are the most widespread and numerous of the mammals, occurring throughout the world. Eutherian mammals are sometimes called placental mammals, because all species possess a complex chorioallantoic placenta that connects a fetus to the mother, allowing for gas, fluid, and nutrient exchange. There are about 4,000 species of placental mammals in 18 to 20 orders with various adaptations for burrowing, flying, swimming, hunting, running, and climbing. In the evolutionary sense, they have been incredibly successful in form, diversity, and abundance. The eutherian mammals are classified in 2 major clades, the Atlantogenata and the Boroitheria. The Atlantogenata include the Afrotheria, e.g., elephants, hyraxes, and manatees, and the Xenarthra, anteaters, armadillos, and sloths. The Boroitheria contain 2 large groups, the Euarchontogliers and the Lorisiatheria. Familiar orders in the Euarchontogliers are the Scandentia, tree shrews, Rodentia, rats, mice, squirrels, porcupines, Lagomorpha, rabbits and hares, and the primates, including humans. Major Lorisiatherian orders include the Perissodactyla, e.g., horses and rhinos, the Cetartiodactyla, e.g., cows, giraffes, pigs, hippos, and whales, the Carnivora, e.g., cats, dogs, and bears, and the Chiroptera, bats and flying foxes. The 2 largest orders are the rodents, 2,000 species, and bats, about 1,000 species, which together constitute approximately 60% of all eutherian species. 29.7 The evolution of primates. By the end of this section, you will be able to do the following. Describe the derived features that distinguish primates from other animals. Describe the defining features of the major groups of primates. Identify the major hominin precursors to modern humans. Explain why scientists are having difficulty determining the true lines of descent in hominids. Order primates of class Mammalia includes lemurs, tarsiers, monkeys, apes, and humans. Non-human primates live primarily 814. Chapter 29. Vertebrates. Access for free at openstax.org. In the tropical or subtropical regions of South America, Africa, and Asia. They range in size from the mouse lemur at 30 grams, 1 ounce, to the mountain gorilla at 200 kilograms, 441 pounds. The characteristics and evolution of primates are of particular interest to us as they allow us to understand the evolution of our own species. Characteristics of primates. All primate species possess adaptations for climbing trees, as they all descended from tree dwellers. This arboreal heritage of primates has resulted in hands and feet that are adapted for climbing, or brachiation, swinging through trees using the arms. These adaptations include, but are not limited to: 1. A rotating shoulder joint, 2. A big toe that is widely separated from the other toes, except humans, and thumbs sufficiently separated from fingers to allow for gripping branches, and 3. Stereoscopic vision, 2 overlapping fields of vision from the eyes, which allows for the perception of depth and gauging distance. Other characteristics of primates are brains that are larger than those of most other mammals, claws that have been modified into flattened nails, typically only one offspring per pregnancy, and a trend toward holding the body upright. Order primates is divided into 2 groups: Strepsirrhini, turned-nosed, and Haplorhini, simple-nosed primates. Strepsirrhines, also called the wet-nosed primates, include prosimians like the bush babies and patos of Africa, the lemurs of Madagascar, and the lorises of Southeast Asia. Haplorhines, or dry-nosed primates, include tarsiers, figure 29.42, and simians, New World monkeys, Old World monkeys, apes, and humans. In general, strepsirrhines tend to be nocturnal, have larger olfactory centers in the brain, and exhibit a smaller size and smaller brain than anthropoids. Haplorhines, with a few exceptions, are diurnal, and depend more on their vision. Another interesting difference between the strepsirrhines and haplorhines is that strepsirrhines have the enzymes for making vitamin C, while haplorhines have to get it from their food. Figure 29.42a. Philippine tarsier. This tarsier, Carlito sarichta, is one of the smallest primates, about 5 inches long, from nose to the base of the tail. The tail is not shown, but is about twice the length of the body. Note the large eyes, each of which is about the same size as the animal's brain. And the long hind legs. Credit. MTOZ, http://creativecommons.org/licenses/by-saw/2.0, http://openstax.org/l/ccsa_2 Via Wikimedia Commons. Evolution of primates. The first primate-like mammals are referred to as proto-primates. They were roughly similar to squirrels and tree shrews in size and appearance. The existing fossil evidence, mostly from North Africa, is very fragmented. These proto-primates remain largely mysterious creatures until more fossil evidence becomes available. Although genetic evidence suggests that primates diverged from other mammals about 85 Maya, the oldest known primate-like mammals with a relatively robust fossil record date to about 65 Maya. Fossils like the proto-primate Placiodopus, although some researchers do not agree that Placiodopus was a proto-primate, had some features of the teeth and skeleton in common with true primates. They were found in North America and Europe in the Cenozoic and went extinct by the end of the Eocene. The first true primates date to about 55 Maya in the Eocene epoch. They were found in North America, Europe, Asia, and Africa. These early primates resembled present-day prosimians such as lemurs. Evolutionary changes continued in these early 29.7. The evolution of primates. 815 primates, with larger brains and eyes, and smaller muzzles being the trend. By the end of the Eocene epoch, many of the early prosimian species went extinct due either to cooler temperatures or competition from the first monkeys. Anthropoid monkeys evolved from prosimians during the Oligocene epoch. By 40 million years ago, evidence indicates that monkeys were present in the New World, South America, and the Old World, Africa and Asia. New World monkeys are also called Platyrrhini, a reference to their broad noses, figure 29.43. Old World monkeys are called Catyrrhini, a reference to their narrow, downward-pointed noses. There is still quite a bit of uncertainty about the origins of the New World monkeys. At the time the Platyrrhines arose, the continents of South America and Africa had drifted apart. Therefore, it is thought that monkeys arose in the Old World and reached the New World either by drifting on log rafts or by crossing land bridges. Due to this reproductive isolation, New World monkeys and Old World monkeys underwent separate adaptive radiations over millions of years. The New World monkeys are all arboreal, whereas Old World monkeys include both arboreal and ground-dwelling species. The arboreal habits of the New World monkeys are reflected in the possession of prehensile or grasping tails by most species. The tails of Old World monkeys are never prehensile and are often reduced, and some species have ischial callosities, thickened patches of skin on their seats. Figure 29.43 A New World monkey. The howler monkey is native to Central and South America. It makes a call that sounds like a lion roaring. Credit. Chavi Talata. Apes evolved from the catarrhines in Africa midway through the Cenozoic, approximately 25 million years ago. Apes are generally larger than monkeys and they do not possess a tail. All apes are capable of moving through trees, although many species spend most their time on the ground. When walking quadrupedally, monkeys walk on their palms, while apes support the upper body on their knuckles. Apes are more intelligent than monkeys, and they have larger brains relative to body size. The apes are divided into two groups. The lesser apes comprise the family Hylobatidae, including gibbons and siamangs. The great apes include the genera Pan, chimpanzees and bonobos, gorilla, gorillas, pongo, orangutans, and Homo, humans. Figure 29.44. Figure 29.44 Primate skeletons. All great apes have a similar skeletal structure. Credit. Modification of work by Tim Vickers, 816 Chapter 29. Vertebrates access for free at openstax.org. The very arboreal gibbons are smaller than the great apes. They have low sexual dimorphism, that is, the sexes are not markedly different in size. Although in some species, the sexes differ in color, and they have relatively longer arms used for swinging through trees. Figure 29.45a. Two species of orangutan are native to different islands in Indonesia: Borneo, P. pygmaeus, and Sumatra, P. abele. A third orangutan species, Pongo tapanuliensis, was reported in 2017 from the Batang Toru forest in Sumatra. Orangutans are arboreal and solitary. Males are much larger than females and have cheek and throat pouches when mature. Gorillas all live in Central Africa. The eastern and western populations are recognized as separate species, G. barangay and G. gorilla. Gorillas are strongly sexually dimorphic, with males about twice the size of females. In older males, called silverbacks, the hair on the back turns white or gray. Chimpanzees, figure 29.45b, are the species considered to be most closely related to humans. However, the species most closely related to the chimpanzee is the bonobo. Genetic evidence suggests that chimpanzee and human lineages separated 5 to 7 Maya, while chimpanzee, Pan troglodytes, and bonobo, Pan paniscus, lineages separated about 2 Maya. Chimpanzees and bonobos both live in Central Africa, but the two species are separated by the Congo River, a significant geographic barrier. Bonobos are slighter than chimpanzees, but have longer legs and more hair on their heads. In chimpanzees, white tail tufts identify juveniles, while bonobos keep their white tail tufts for life. Bonobos also have higher pitched voices than chimpanzees. Chimpanzees are more aggressive and sometimes kill animals from other groups, while bonobos are not known to do so. Both chimpanzees and bonobos are omnivorous. Orangutan and gorilla diets also include foods from multiple sources, although the predominant food items are fruits for orangutans and foliage for gorillas. Figure 29.45 Lesser and Great Apes. This white-cheeked gibbon, A, is a lesser ape. In gibbons of this species, females and infants are buff and males are black. This young chimpanzee, B, is one of the great apes. It possesses a relatively large brain and has no tail. Credit A. Mac. Credit B. Modification of work by Aaron Logan. Human evolution. The family Hominidae of order Primates includes the hominoids, the great apes and humans. Figure 29.46. Evidence from the fossil record and from a comparison of human and chimpanzee DNA suggests that humans and chimpanzees diverged from a common hominoid ancestor approximately 6 million years ago. Several species evolved from the evolutionary branch that includes humans, although our species is the only surviving member. The term hominin is used to refer to those species that evolved after this split of the primate line, thereby designating species that are more closely related to humans than to chimpanzees. A number of marker features differentiate humans from the other hominoids, including bipedalism or upright posture, increase in the size of the brain, and a fully opposable thumb that can touch the little finger. Bipedal hominins include several groups that were probably part of the modern human lineage: Australopithecus, Homo habilis, and Homo erectus, and several non-ancestral groups that can be considered cousins of modern humans, such as Neanderthals and Denisovans. Determining the true lines of descent in hominins is difficult. In years past, when relatively few hominin fossils had been recovered, some scientists believed that considering them in order, from oldest to youngest, would demonstrate the course of evolution from early hominins to modern humans. In the past several years, however, many new fossils have been found, and it is clear that there was often more than one species alive at any one time and that many of the fossils found, and species named, represent hominin species that died out and are not ancestral to modern humans. 29.7. The Evolution of Primates 817 Figure 29.46 Hominin Phylogeny. This chart shows evolutionary relationship among hominins and hypothesized relation to modern humans. Still debated phylogeny position. Very early hominins. 3 species of very early hominids have made news in the late 20th and early 21st centuries: Ardipithecus, Sahelanthropus, and Orrorin. The youngest of the 3 species, Ardipithecus, was discovered in the 1990s, and dates to about 4.4 Maya. Although the bipedality of the early specimens was uncertain, several more specimens of Ardipithecus were discovered in the intervening years and demonstrated that the organism was bipedal. 2 different species of Ardipithecus have been identified, A. ramidus and A. cadaba, whose specimens are older, dating to 5.6 Maya. However, the status of this genus as a human ancestor is uncertain. The oldest of the 3, Sahelanthropus chidensis, was discovered in 2001–2002 and has been dated to nearly 7 million years ago. There is a single specimen of this genus, a skull that was a surface find in Chad. The fossil, informally called tumai, is a mosaic of primitive and evolved characteristics, and it is unclear how this fossil fits with the picture given by molecular data, namely that the line leading to modern humans and modern chimpanzees apparently bifurcated about 6 million years ago. It is not thought at this time that this species was an ancestor of modern humans. A younger, circa 6 Maya, species, Orrorin tuganensis, is also a relatively recent discovery, found in 2000. There are several specimens of Orrorin. Some features of Orrorin are more similar to those of modern humans than are the Australopithecines, although Orrorin is much older. If Orrorin is a human ancestor, then the Australopithecines may not be in the direct human lineage. Additional specimens of these species may help to clarify their role. Early hominins. Genus Australopithecus. Australopithecus, southern ape, is a genus of hominin that evolved in eastern Africa approximately 4 million years ago and went extinct about 2 million years ago. This genus is of particular interest to us as it is thought that our genus Homo, 818 Chapter 29, vertebrates access for free at OpenStax.org. Evolved from a common ancestor shared with Australopithecus about 2 million years ago, after likely passing through some transitional states. Australopithecus had a number of characteristics that were more similar to the great apes than to modern humans. For example, sexual dimorphism was more exaggerated than in modern humans. Males were up to 50% larger than females, a ratio that is similar to that seen in modern gorillas and orangutans. In contrast, modern human males are approximately 15 to 20% larger than females. The brain size of Australopithecus relative to its body mass was also smaller than in modern humans and more similar to that seen in the great apes. A key feature that Australopithecus had in common with modern humans was bipedalism, although it is likely that Australopithecus also spent time in trees. Hominin footprints, similar to those of modern humans, were found in Laetoli, Tanzania and dated to 3.6 million years ago. They showed that hominins at the time of Australopithecus were walking upright. There were a number of Australopithecus species, which are often referred to as Australopiths. Australopithecus inamensis lived about 4.2 million years ago. More is known about another early species, Australopithecus afarensis, which lived between 3.9 and 2.9 million years ago. This species demonstrates a trend in human evolution: the reduction of the dentition and jaw in size. A. afarensis, figure 29.47a, had smaller canines and molars compared to apes, but these were larger than those of modern humans. Its brain size was 380 to 450 cubic centimeters, approximately the size of a modern chimpanzee brain. It also had prognathic jaws, which is a relatively longer jaw than that of modern humans. In the mid-1970s, the fossil of an adult female A. afarensis was found in the Afar region of Ethiopia and dated to 3.24 million years ago, figure 29.48. The fossil, which is informally called Lucy, is significant because it was the most complete Australopith fossil found, with 40% of the skeleton recovered. Figure 29.47 Australopithecine and modern human skulls. The skull of A. Australopithecus afarensis, an early hominid that lived between 2 and 3 million years ago, resembled that of B. modern humans but was smaller with a sloped forehead, larger teeth, and a prominent jaw. 29.7. The evolution of primates 819 figure 29.48 Lucy. This adult female Australopithecus afarensis skeleton, nicknamed Lucy, was discovered in the mid-1970s. Credit 120 Wikimedia Commons. Australopithecus africanus lived between 2 and 3 million years ago. It had a slender build and was bipedal, but had robust arm bones and, like other early hominids, may have spent significant time in trees. Its brain was larger than that of A. afarensis at 500 cubic centimeters, which is slightly less than one-third the size of modern human brains. Two other species, Australopithecus barralghazali and Australopithecus gari, have been added to the roster of Australopiths in recent years. A. barralghazali is unusual in being the only Australopith found in Central Africa. A dead end. Genus Paranthropus. The Australopiths had a relatively slender build and teeth that were suited for soft food. In the past several years, fossils of hominids of a different body type have been found and dated to approximately 2.5 million years ago. These hominids, of the genus Paranthropus, were muscular, stood 1.3 to 1.4 meters tall, and had large grinding teeth. Their molars showed heavy wear, suggesting that they had a coarse and fibrous vegetarian diet as opposed to the partially carnivorous diet of the Australopiths. Paranthropus includes Paranthropus robustus of South Africa, and Paranthropus ethiopicus and Paranthropus boisei of East Africa. The hominids in this genus went extinct more than 1 million years ago and are not thought to be ancestral to modern humans, but rather members of an evolutionary branch on the hominin tree that left no descendants. Early hominins. Genus Homo. The human genus, Homo, first appeared between 2.5 and 3 million years ago. For many years, fossils of a species called H. habilis were the oldest examples in the genus Homo, but in 2010, a new species called Homo gautengensis was discovered and may be older. Compared to A. africanus, H. habilis had a number of features more similar to modern humans. H. habilis had a jaw that was less prognathic than the Australopiths and a larger brain, at 600 to 750 cubic centimeters. However, H. habilis retained some features of older hominin species, such as long arms. The name H. habilis means, handy man, which is a reference to the stone tools that have been found with its remains. Link to learning. Watch this video about Smithsonian paleontologist Brianna Pobiner explaining the link between hominin eating of meat and evolutionary trends. Click to view content, https://www.openstax.org/l/diet_detective 820 Chapter 29, Vertebrates access for free at openstax.org. H. erectus appeared approximately 1.8 million years ago, figure 29.49. It is believed to have originated in East Africa and was the first hominin species to migrate out of Africa. Fossils of H. erectus have been found in India, China, Java, and Europe, and were known in the past as Java man or Peking man. H. Erectus had a number of features that were more similar to modern humans than those of H. habilis. H. erectus was larger in size than earlier hominins, reaching heights up to 1.85 meters and weighing up to 65 kilograms, which are sizes similar to those of modern humans. Its degree of sexual dimorphism was less than in earlier species, with males being 20 to 30% larger than females, which is close to the size difference seen in our own species. H. erectus had a larger brain than earlier species at 775 to 1,100 cubic centimeters, which compares to the 1,130 to 1,260 cubic centimeters seen in modern human brains. H. erectus also had a nose with downward-facing nostrils similar to modern humans, rather than the forward-facing nostrils found in other primates. Longer, downward-facing nostrils allow for the warming of cold air before it enters the lungs and may have been an adaptation to colder climates. Artifacts found with fossils of H. erectus suggest that it was the first hominin to use fire, hunt, and have a home base. H. erectus is generally thought to have lived until about 50,000 years ago. Figure 29.49 Homo erectus. Homo erectus had a prominent brow and a nose that pointed downward rather than forward. Humans. Homo sapiens. A number of species, sometimes called archaic Homo sapiens, apparently evolved from H. erectus starting about 500,000 years ago. These species include Homo heidelbergensis, Homo rhodesiensis, and Homo neanderthalensis. These archaic H. sapiens had a brain size similar to that of modern humans, averaging 1,200 to 1,400 cubic centimeters. They differed from modern humans by having a thick skull, a prominent brow ridge, and a receding chin. Some of these species survived until 30,000 to 10,000 years ago, overlapping with modern humans. Figure 29.50. 29.7. The evolution of primates. 821. Figure 29.50 Neanderthal. The Homo neanderthalensis used tools and may have worn clothing. There is considerable debate about the origins of anatomically modern humans or Homo sapiens sapiens. As discussed earlier, H. erectus migrated out of Africa and into Asia and Europe in the first major wave of migration about 1.5 million years ago. It is thought that modern humans arose in Africa from H. erectus and migrated out of Africa about 100,000 years ago in a second major migration wave. Then, modern humans replaced H. erectus species that had migrated into Asia and Europe in the first wave. This evolutionary timeline is supported by molecular evidence. One approach to studying the origins of modern humans is to examine mitochondrial DNA, mtDNA, from populations around the world. Because a fetus develops from an egg containing its mother's mitochondria, which have their own, non-nuclear DNA, mtDNA is passed entirely through the maternal line. Mutations in mtDNA can now be used to estimate the timeline of genetic divergence. The resulting evidence suggests that all modern humans have mtDNA inherited from a common ancestor that lived in Africa about 160,000 years ago. Another approach to the molecular understanding of human evolution is to examine the Y chromosome, which is passed from father to son. This evidence suggests that all men today inherited a Y chromosome from a male that lived in Africa about 140,000 years ago. The study of mitochondrial DNA led to the identification of another human species or subspecies, the Denisovans. DNA from teeth and finger bones suggested two things. First, the mitochondrial DNA was different from that of both modern humans and Neanderthals. Second, the genomic DNA suggested that the Denisovans shared a common ancestor with the Neanderthals. Genes from both Neanderthals and Denisovans have been identified in modern human populations, indicating that interbreeding among the three groups occurred over part of their range.